So because seeds were limited to around 100 per variety, we planted 2 seeds together and then thinned. So we ended up with about 50 plants per variety. That's not taken into account if we lost a few. So we planted our trials on a half an acre field. And most years we planted our trials next to larger melon trials in the Yuma Ag Center. So this facilitated the volunteer bees to come and pollinate our crop. So we never had to rent bee boxes to have any pollinators because of the amount of fields around our trials. And here's some images of some of the planting. So basically, unlike the commercial melon growers, we would plant our trials by hand because of the limited number of seed. So essentially all I would do, I would lay down a measuring tape and I would go by every 2 feet and drop a couple seeds, about 3/4 of an inch. Normally, we could have used a hopper, which would have, I guess it would have simulated more of a commercial manner of planting. But because we were limited to seed, often hoppers may break seeds. And so we wanted to make sure that the limited number we were working with was used to the fullest extent. So for the first year, we were given 6 varieties to work with. Second year, well, and those first 6 varieties were already commercial melons, I believe. The second year we had those 6, and then we had some trial hybrid varieties that were bred by Dr. Kevin Crosby. And the following year, in year 3, we were given 6 Syngenta varieties and I believe 8 trial hybrid varieties for a total of 16. And year 4, it was a total of 18 varieties. And each year we harvested about a dozen to go to both Texas A&M for the consumer studies and to Sadhana, Dr. Ravishankar for the prevalence studies. And this is just to show you guys, we had in our trials, of course, we had multiple varieties. On the left, you can see the honeydew, and on the right is a cantaloupe. And I'll talk about some of the complications that arose with having different varieties in the same trial. So as far as irrigations and chemical applications and pesticide applications, so for drip, we use drip irrigation for the first 4 or 5 days while the plants germinated, and this was a surface drip irrigation. After we had our plants germinate, we would remove the surface drip, and that's when we would begin to use the irrigation, or the drip irrigation that was set about 6 inches below the seed line. In some instances, it wasn't too often, but furrow water was used, and in some instances as well, some fertilizers were applied through that furrow water. This is mostly the Yuma Ag Center's farm practices. It's not so much the commercial practices, but in some instances that was done. Insecticides, pesticides, and fungicides were applied periodically, or in instances where pest pressure or disease pressure was high. So unlike a commercial setting, they have what is an economic threshold. When you're dealing with multiple, like 20 plus acres of crops, they'll have a PCA go out and write a recommendation, because applying insecticides or pesticides over such a large area tends to be pretty expensive. We didn't have those complications because it was a very small trial, so we were able to periodically apply insecticides and pesticides as needed. And here's a picture of some germinated melon crops. So this is probably about the time that we would remove that drip tape. And as you can see, there's 2 crops. As I said, I'd planted double seeds. So once we remove the drip tape, I would go through and I would thin out those crops, leaving the single crop in its stead. So some of the complications we encountered, as I mentioned earlier, we had multiple varieties in the same trial. So that led to the maturation of each variety being at different states. So that complicated a bit the harvest time. Some of the other complications were pests such as whiteflies, which we encountered every year. It wasn't a significant problem, as I said, because we were able to periodically apply insecticides. But as soon as the climate started to warm up, we would always see whiteflies under the canopy leaves. Powdery mildew was also found. It wasn't a big deal, but we found it pretty often in the field trials. And by the way, this is caused by a fungus called Posidora xanthi. But like I said, since we're able to apply the fungicides periodically, it wasn't much of a big deal. So one instance where we did have a pretty major setback was during the third year of planting. We had consecutive rainstorms beginning probably at the end of February into early March. So for 3 whole weeks, we had a large rainstorm. And for the Yuma area, even a 1/4 inch of rain is pretty significant. So that delayed our planting by a whole 3 weeks, in turn delaying our harvest time. So we would rather, if everything went according to plan, we would harvest our crops early June, mid-June, before the temperature started to increase too much. And in this case, we had to push it closer to the beginning of July. By then, we have temperatures reaching over 110 degrees. So the fruit doesn't tend to do too well in those temperatures. Vine decline caused by root infecting ascomycete, Monosporascus, Cannonballus was something that was quite a significant problem in a few years of our trials. I have a few images of that, which I'll explain. And as everybody knows, we also experienced some complications due to the COVID-19 pandemic. We had a lot of our staff working remotely. Myself, I was working remotely, so I didn't have the same opportunities to scout and maintain the field as I normally would working from the office. Here I just included a picture of powdery mildew, just to give you guys an idea of what that looks like. As I said, this wasn't such a huge complication because we were able to deal with that in a timely manner before it got out of control. And here's some images of what vine decline would look like in our trial. So off to the top left corner, you can see what a healthy canopy is supposed to look like. And over here, more to the lower center, is some crops affected by vine decline. So unless you do soil testing, you're not really aware that the microbes or the organisms present in the soil. You only start to notice maybe 2 to 3 weeks before harvest time when the canopy starts to die off. And this creates a pretty significant complication because you don't want to harvest 2 or 3 weeks before the crops or the fruit's ready. And in many instances, we were really looking for the last bits of fruit we could find to complete the amount of fruit that we needed to send both to Texas and Tucson. And here's also another picture of some severe vine decline. So as you can see, the canopy is basically completely gone. The fruit's completely exposed to the sun, which causes burns. It starts to rot, and that really limits the amount of fruit that we have to harvest to send to both locations. But we were able to do that even with these complications. We were able to send the amount of fruit that we needed to get to both places. So for shipping, melons were harvested and shipped to both Texas and Tucson, as I said. About a dozen per variety for each year was shipped to both locations. The number of fruits shipped to each location varied each year. Usually, as I said, 10 to 15, usually a dozen. Tucson also received a shipment of commercially grown melons from the Yuma area during the fall growing season. The number per year also varied, but the total commercial melons shipped over the 4-year period was well over 1,000. And there were no commercial melons shipped to Tucson during 2020 due to the COVID pandemic. This is a shipment of melons that was sent to Texas. So some of the things, our first year, we didn't realize that a lot of the fruit during the shipment gets jostled. Some of it ends up broken. So we had to reship some of that stuff our first year. Our second year, we learned that we needed to set some bumpers. So we tried using bubble wrap, which worked. It worked well enough, but since some of the melon rinds are very rough, it tends to pop that bubble wrap, making it not so effective. But we found what worked really well was pool noodles. They're very inexpensive and firm, so that worked well in keeping the melons in place and allowing them to arrive in one piece to where they need to go. So for outreach, a field day was held in the Yuma Agriculture Center during the first year of the project. Commercial growers attended and discussed the goals of the project with the project leads. This is where they were able to voice some of their concerns, or as well as the project leads were able to ask the commercial growers what it is exactly that they wanted to see in the project itself. I included a picture of the field day here. As you can see from left to right, Dr. Ravishankar, Dr. Patil, Dr. Crosby, and Dr. Jafan, and my boss, Paul Brierly in the front, and in the audience is other commercial growers. And after the discussion, we all went out and toured the field. So this is our first year trials. And of course, the acknowledgements. And that concludes that presentation. So if you guys have any questions, feel free to ask.
Anybody has any questions for Martin? You want to check the chat to see if there is on the top? Nothing in the chat?
Nothing in the chat.
So any questions anybody has for Martin? So I have one question, Martin, while presenting. So with the wine decline, you did mention, that the only way to test is by testing the soil.
Mm-hmm.
So let's say 1 year, the growers find that there is wine decline in that field. What do they do for the following year? Do they plant that field and plant somewhere else? They never go back to that field, right? I mean,
so
how do they?
Do you mean from the commercial perspective?
From
or
the
from
commercial perspective,
so
once they see that one field has had vine declining disease,
Mhm.
Next year, what do they use the land for? I mean, or do they not plant it?
Well, it depends, because from a commercial perspective, if you lose, so Sadhana asked, what would a commercial grower do if they found that they have vine decline in one of their melon fields? Would they continue to plant cantaloupe in that field the consecutive years, or would they plant something else? And from what I understand, because it's similar to how lettuce growers do with fusarium, although the returns are different for both those crops. But depending how bad it is in the field, they're usually willing to take the loss, because they'll make back that their investment, depending on how severe it is. They'll if it's too severe, they might not plant cantaloupe in that area. They might move it to a different location. And I'm not sure, I couldn't tell you exactly which crops, but like with fusarium, planting broccoli in a field that is infected is going to keep that organism. I don't know if it functions as a host. It doesn't affect the crop itself, but it maintains the crop. It maintains it in the soil, so it'll keep its presence there. So I assume that would come down to an economic question. So whether the losses would be more than they can recuperate by planting in that field.
And how many years? I mean, this continues. If the vine decline has been there, is it like that for like 7 years?
Yeah, I imagine it would be several years. It's a pretty hardy spore. It can survive dormant in the soil for a while. So for us, we had it in an area. They just moved us to a different area. Turns out that area also had it, and they were unaware of it. So yeah, it tends to linger around for a long time.
It for a long time.
Yeah.
Okay. Yeah, but while Dr. Crosby is sharing it, I just missed the introduction part of him. I think he's the only breeder on melons left in the country now. So just to get you an idea, and also it's a good continuation of what Porchas presentation on that disease. And Dr. Crosby has done some good work on that particular resistance to that particular pathogen. So he will talk about that along with some of the varieties he's coming out with this project. Go ahead, Dr. Crosby.
Oh, okay, thanks. Yeah, the two options to deal with that vine decline are methyl bromide, which is now off the market, and there's a fungicide called Cannonball. It's very expensive, but it's restricted use because they don't want the fungus to develop resistance. So those are some options, or expensive options. So a better option for us is developing resistance. So first, I'd like to acknowledge the grant and the people that helped me. There's been a lot of work on breeding resistant melons against a lot of diseases at A&M Research Center, Wessico, since 1941. The first major disease was downy mildew, and then powdery mildew, fusarium, and subsequently in the last 25 years has been Monosporascus vine decline, which is, Martin says, is in the soil and will stay in the soil for many, many years in the absence of some strong fumigant. So a lot of varieties were developed, and that's been our goal, is to develop disease-resistant cantaloupe varieties. There's not, as Dr. Patil mentioned, there's not any public cantaloupe breeders left in the US. They're all in the private industry. And the US market's been shrinking. So we're trying to come up with some varieties that taste better. And so we've done a lot of evaluation of material, not just for resistance, but also has to look pretty good and taste good. So I think Dr. Singh will talk about that. So we have brought resistance from these wild type melons that come from Africa and India. They're small, they're sugar-free. They don't have the same texture or behavior, but they possess remarkable disease resistance. So in fact, here's a melon we developed some years back, which has some DNA segments from this wild melon for resistance to mildew and vine decline. But subsequently, this was partially resistant to vine decline, but has good quality. Subsequently, we decided we needed higher levels of resistance to that vine decline. As Martin mentioned, when it gets in, builds up in the soil, it's really difficult for the roots to survive. Okay, it eats away the roots. They call it also root rot, and then the plants collapse, typically before they're mature, so you lose the crop. This is what it looks like, and you've already invested a lot in the crop if you're the grower, and then a week or two before harvest, it collapses, so you really lose everything that you've invested. So that's terrible. It would be better if the plants died right when they, germinated, and at least you wouldn't waste all the time, fertilizer and water. So we identified resistant material going all the way back to when I was a PhD student, just by screening material from all over the world. We wanted to improve the resistance to this disease. We also wanted to improve the root systems. A lot of the cantaloupe cultivars had small, weak root systems, and that also exacerbates drought stress problems, susceptibility to this disease and other diseases, and it affects quality because the root systems need to be healthy and vigorous to bring all the water and the fertilizer from the soil into the crop. So we did a lot of screening and excavating roots. Incidentally, at Wessico Center, because of 60 or 70 years of melon cultivation, we have a soils very infested with the Monosporascus and fusarium and other things. So we conducted greenhouse inoculations just with that fungus, Monosporascus, so we could avoid interaction with other potential pathogens. We verified some resistance. So here you have red is inoculated, white is not. As you can see, the top one is much more resistant when inoculated compared to the bottom line. These are just some breeding lines. Here's an example of where under heavy inoculation, the commercial variety here just completely dies from the vine decline. The roots are completely rotted. And our breeding line, which utilizes a resistant type of melon from India in its breeding, survives. And the reason is because the roots are not rotted. And you can see when we wash the sand off and look at the roots. I have a student who's been working on this right now, and he's found some interesting profiles of amino acids and things in some of this resistant type. So could be that, you know, it's creating some chemicals in its roots, potentially providing it with resistance. We're looking into that more. The resistance is fairly dominant, which is good. I'll mention that in a minute. So that makes it easier from a breeding perspective. So here's a resistant thing from India, which doesn't taste good. It looks kind of like a pumpkin. So anyway, after a lot of breeding and screening and inoculating and back crossing, we've come up with cantaloupes. Excuse me. And we've also, you know, improved the flesh quality a lot. And so the new hybrids we're developing have these cantaloupe breeding lines as parents that have this resistance and, you know, cantaloupe attributes. During this process of 22 years that we've doing this, we've developed about 2,000 breeding lines, and some of them are cantaloupes and some of them are other things, and some of them are honeydews, and some things are kind of in a category all by themselves. So that might lead to something new, maybe a new type of melon. So we've been testing hybrids, and my student and I have done some genetic studies. And what turns out is that there's a strong dominant component to this resistance, which is very good, because from a breeding standpoint, that makes it a lot easier. Probably could be 2 or 3 dominant genes, or at least there's a major dominant gene and maybe some other modifying type genes. So the heritability was pretty good in these family. Moderate to heritability in the narrow sense, and the broad sense higher. So we determined that there's a strong dominant component, but there's also an additive effect. So if both parents are resistant, you get kind of this additive effect, as opposed to just one parent being resistant. But even with one parent resistant, highly resistant, the hybrid can still be resistant, as we found in some of our hybrids we've developed. So this is positive development, because like I said, methyl bromide's off the market is dangerous, and that the one fungicide for Monosporascus is very expensive, and it's restricted use, highly restricted, because they don't want it to become, the fungus to become resistant. So during these last couple of years, we've taken our best quality resistant lines and crossed them together to create 40 new hybrids for field trials. And Martin mentioned about 15 or so that we trialed in Yuma. We trialed more even in Wessico and Uvalde here. And the key is to find out which ones are resistant, but also have a desirable cantaloupe, because there are a lot of bad tasting melon cultivars on the market already. So I don't really want to just develop something resistant that tastes bad. So here's at Wessico, where we have a lot of the Monosporascus. We also have some other diseases, but that's the main one. And the vines are very strong. You can see that's a reflection of a strong root system. We found there's a definitely correlation between a healthy root system and a strong vine that is able to develop large fruit. So oftentimes we got very large size fruit, 9 or even 6 size fruit. 6 is too big. So what we're targeting is 9 to 12. That's what the industry wants. And these are 3 of the best hybrids for resistance to the pathogen, Monosporascus plus powdery mildew resistance, which is common in Texas and Arizona and California. But also have the size is okay, the shape is okay. They have a net that looks like a normal cantaloupe, and they have interior quality that's good, orange, thick flesh. The bricks tends to be good. Usually when bricks is good at Wessico, which is a very hot, humid place, even at nighttime, it's hot and humid. That means the bricks is typically going to be even better if you put it out in the desert, like California or Arizona or Pecos region of Texas. So the other thing we have to consider is firmness. And so like this TH6 has a very good flavor and it's very productive, but in some locations, particularly Wessico, it wasn't very firm. That's not very firm. This is very firm, number 9. And naturally, even though it's very firm and productive, it had a little bit less bricks and a little bit less flavor than the number 6. And then this was kind of a in between. This 13 we're releasing, and it has a very good flavor in bricks, and it has pretty firm fruit in most locations. Its main problem is inconsistent size. So like at Wessico, size was okay, size 12. At Uvalde, it was more of a desert climate. It was very small, size 15 or 18, which is not acceptable. Now, we grew this in California, and I looked at it, and it was nice. It was size 12, size 9. So it's very sensitive to the location. So I think it will be a good melon for certain locations. It has a very good flavor. And these are just some of the data we have from College Station. Is not a great place for melons because it rains a lot, but we put them there just to see the effect on quality and firmness. And we found even in that environment, some of our, like this is 13, 52 times 96, and it's firm and it had high bricks even in that environment. So that's a good sign because that's kind of a semi-tropical, humid, almost like Louisiana or East Texas type climate. It's not ideal for cantaloupes. So that's a pretty good indicator that it's pretty adapted to high heat and humidity and stress. And then these are the one. This is the number 13, and I think I have a picture of number 6. That's the other one we're releasing, just because number 6 is very, very productive, and at least under Texas conditions, and it has a large fruit which they like. Here's number 6. So this one of all the ones was the highest yielding of the cantaloupe types, and has a very strong resistance to the pathogen, to the vine decline, to mildew. Produces a large, attractive fruit, and it has a good flavor with high bricks, consistently high bricks. In fact, it's always always the highest bricks. However, it's not the firmest melon, so it's going to be a situation where it's going to need to be trialed in specific locations to see if it's too firm or if it's too soft or if it's acceptable. And then this is just an example of the resistance we introduced from that wild type to the Monosporascus. So this is from our Monosporascus infested soil at Wessico. And you can see how healthy this is at maturity. After we've harvested the fruits, we dug up the vine, and this is how healthy the roots are. So that helps the plant to be strong, no question about it. Even at the end of the season, the plant is still surviving and growing. The only question is, can we find just the right cultural practices and the ideal locations for this particular melon so it's not too soft? It's not soft in every location, but Wessico is kind of a tough location because of the high humidity. So I'm hoping to get this trialed more out in the desert. And so, like I said, in California, this looked good, but it was smaller, and I was somewhat surprised because in Texas, it tends to be a little bit oversized. But in California, the quality was fine, but it was a smaller melon. So I guess it really depends on the environment and the market. So we'll see. We're releasing these guys because they have good flavor and they have vine decline resistance. And then it'll be up to the growers in specific regions to see if they're adapted to their specific soil and their specific growing conditions. And that's all that I have.
So, I have one question, Dr. Crosby. So, I know you were mentioning the size, the firmness, and some of the, desirable attributes in a melon. So, what will your, most ideal TSS size and firmness would be, for a melon that you're trying to create, or what the most ideal range would be for the best, type of melon?
Well, typically they want size 9 and they want firmness probably in excess of 70 newtons. So the ones that are 90, 95, like the ones I showed is more than firm enough. And then the color is important and thick flesh. They don't want a large open cavity because that's just not desirable.
Oh,
So they don't want too long of a melon that's like a football. They want a melon that's a little more a little rounder, and it doesn't have to be perfectly round, but just not like about this looking melon. The net they like for a cantaloupe, they like this type of net. Now for the newer melons, like the long shelf life, they don't seem to care that much about the net. It's not that big of a deal.
Oh, okay. And how much should the TSS be? I mean, or the bricks?
Well, we like to say that 10 is the minimum that's acceptable, and 12 is good. Now, when you get above 12, most people will say, wow, that's outstanding. That's really amazingly sweet, like 13 or 14. But I think 12 is, everybody's satisfied when they have 12.
Okay. Thank you.
Okay.
And I think there is a question in the chat. Where I can get the seeds for trial.
Oh yeah, I can send out small seed samples of seed. Well, actually, that TH6 we have a fair amount of seed. We tried to produce some seed in Thailand, but I just got notification that they pretty much screwed that up. So, but I have breeder seed. I can send out like 100 seed or a couple of hundred seed for people if they want to plant a row and take a look at it. And hopefully by later this year we'll try to produce the seed again with a commercial seed company.
Again, while he's uploading this presentation, I just like to stress that Dr. Singh, again, as you heard from Dr. Crosby, I think he is really doing a great job in breeding melons and other crops. But melon, I think out of this project, we'll have several hybrids suitable for disease resistance along with some of the health properties. So what you're going to hear now is Dr. Jashbir Singh, who is a postdoc since from 5 years or 7 years working for me. But in this project, he's been working for 5 years. He's the main person involved in phytochemical analysis of all the melons we're receiving from different states. So when breeder Dr. Crosby sends the melons, and he also comes to our center and work on basically selecting the melons based on some of the health properties. So Dr. Singh will talk about those things now.
So good afternoon, everyone. So welcome to the presentation. And first of all, I'd like to let me thanks to the workshop organizer to give me opportunity to present my work on melon. And the title of my presentation is Chemical Profiling of Muskmelon Cultigen Harvested from Different Locations. So the goal of this study was to understand the effect of growing location on the accumulation of the phytochemical.
Dr. Singh, can you be nearby, speak a little bit, because I think they may not be able to hear well.
Okay, hello, you can hear?
Yeah.
So the goal of this study was to understand the effect of growing location on the accumulation of phytochemicals such as carotenoids, amino acid, sugar, and volatile in the newly developed hybrid by the Dr. Kevin Crosby. And so melons are belong to a Cucurbitaceae family, and they are basically the odorous variety and inodorous variety. So cantaloupes are mainly the netted and aromatics in nature, and while the honeydews are the non-netted melons, and which are widely accepted by the consumer for their taste and sweetness. So nutritional composition of melon. So melons are basically the rich source of carotenoids, especially beta carotene, and beta carotene is basically the precursor for the vitamin A, which has a good for the human health eyes. And it has also a good amount of sugar, vitamin C, and dietary fiber. Dietary fiber has helped in improve the gut health, and it also has a good source of volatile flavonoid and phenolic compounds. And health benefit, melons are shows very strong antioxidant activity. They have an anti-inflammatory activity also, and it also improves the cardiovascular health and stimulate the immune system, and has some studies also shows that it has an anti-tumor activity. So these are some published literature on the variation of beta carotene in different melon cultivar, which harvested from the Texas UArizona basically. And you can see the Western Shepherd F339, which has a higher beta carotene content as compared to the other commercial variety like Tuscan Davensi, TTDV, and long shelf life Harper type STIG. And out of the cantaloupe and honeydew, cantaloupe has a higher beta carotene content as compared to the honeydew. And to some extent, orange cassava OC 162, 164 also show some amount of the beta carotene content, but their contents are lower than the traditional cantaloupes. So, and in other study, this study involved the scanning of the volatile and carotene profile in the large genetic breeding of the melons. And the output of the highlight of this study is that the author detected a huge amount of untrapped aroma diversity in the melon germplasm, especially in the non-commercial varieties. So therefore, in our study, we harvested melon from the 4 growing locations: Texas, California, Arizona, Indiana, and we grow then all the melons were grown and shipped to the Texas A&M University. So the We harvested melon in like that. We harvested from the one plant, we harvested melon. So same fruit will take, it go to the sensory analysis and go to the genetic and genomic analysis and phytochemical analysis. And from the fruit from the same plant also go to the food safety to the Arizona, where people do the food safety of the melon. So here we try to get basically the fruit from the same plant or same fruit. We do sensory and phytochemical together and fruit safety together. So these are the some cultigens we grow in all the 4 locations: TH1, TH5, TH6, TH9, 10, 11, 12, and 13 and 14. So these are the various photos of different cultigens. And this is basically how we put the processing. This is in processing. When we receive the melon from the different growing location, we first weigh them. We cut into two halves. Then we take the color reading. After color reading, we take the sample for the sensory analysis. And after that, this sample taken by the other scientists for the genetics and genomic analysis. And later we cut out and blend the sample and take sample for the phytochemical analysis. So sample preparation, just the simple, we take the melon, we blend them, then we do add solvent, different solvent depending upon the interested phytochemicals, and then we do homogenizations, then we do sonication, then after number of steps, we do HP analysis. So overall, you can see that the total soluble solid. So this shows that the total soluble solid is are the important parameter for determining the ripeness of fruit. So generally, you can see in the present result that TSS content vary from 7 to 13 Rics, and generally the melon seed from the Texas, California, and Arizona have a high Rics content around more than 10, except the Indiana variety, Indiana hybrids. They have a lower level of TSS content, and generally they are less than 8.5. All the hybrid shows the less content of total soluble solid. So this is a typical HPLC analysis of sugar chromatogram. And you can see the HPLC chromatogram of the standard showing sucrose, glucose, and fructose. And the bottom, you can see the methanol extract showing the sucrose, glucose, and fructose. So from there, we had done the HPLC analysis, and then later we quantify the content. So sucrose basically it was high in a TS variety, which harvested from the Texas, and TH5, and also a TH5 variety from the Texas and California. And while the lowest level of the sucrose was observed in Indiana fruits. Similarly, the glucose has a highest found in TS13 and from the Texas location, and lowest in and again the lowest in the Arizona fruit in this TS13 shows. And level of fructose. Fructose also followed the similar trend as glucose, and there's no significant difference observed in the TH5 and TH1 from the fruit of Texas, Indiana, and California. And TS13 from the Texas has the highest fructose level as compared to the fruit harvested from the Arizona location. So overall, the total sugar content was observed good in TH1, TH5, TH6, and TH13 had from the Texas location have a higher total sugar content. This slide represents the chemical structure of the beta carotene, which contribute to the major carotenoids in the Cantaloupe, and there was a significant variation on the level of the beta carotene harvested from the different locations. You can see that all the fruit harvested from the Indiana, they have a low level of beta carotene, while the fruit harvested from the other location like Arizona and Indiana, they have a higher level of beta carotene, significantly higher level of beta carotene. And while the TH13 from the Texas has a high level as compared to the Arizona, and their content is approximately 330 microgram per gram. So this is a typical HPLC chromatogram of the amino acid, and you can see the peak 3 and peak 12. They represent they are the major amino acid we observed in the melon, and they are correspond to the glutamine and the GABA. So glutamine, both glutamine and GABA are the neurotransmitter, and they are very important and essential for the brain metabolism and their function. And later in the cycle, glutamine also convert to the GABA. So here we observe that the TH1, TH5, and TH12 from the Arizona location had high glutamine content compared to the other locations, while GABA content similarly TH1, TH5 had a higher GABA in Arizona location, while TH6, you can see here in the Texas location, and TH9 also from the Texas location, TH10 and TH11 also from Texas location has a high GABA content. So the level of citrulline. So citrulline, you can see the sample harvested from the Arizona, like TH1, TH5, and TH12 also has a high citrulline content, and TH6, while in TH6, high citrulline content present in the Texas location. So citrulline is a non-proteaceous and non-essential amino acid, but it acts as a signaling molecule in various metabolic pathways. An accumulation of citrulline in plant is beneficial under stress conditions such as drought, high salt conditions, and it also acts as a reactive oxygen species. While in human, citrulline improves cardiovascular health and enhance the sport performance by reducing fatigue and stress. And citrulline, along with arginine, they help involve in the nitric oxide production, which help in the improvement of cardiovascular health. So you can see also arginine also high in some locations, but they are not so very higher level. So similarly, now we move to the volatile section. So volatile generally we analyze by the GCMS, and we generally use 1 gram of sample with 1 ml of 30 ml of NaCl solution and 5 microliter of internal standard. So GCMS condition, we generally use, in the present study, we use fiber with divinyl benzene carboxyl polydimethylsilicon fiber, and column is RT-VEX column for the suppression of volatile compound, GC injector for held for 225 degrees Celsius, and the temperature of mass line and ion source is 280 and 285 respectively. So generally overall, if you look at the volatile profile of the muskmelon, esters are the major contribute for the volatile and aldehyde, followed by aldehyde, alcohol, and others like sulfide, terpenes, thioesters. These are the reported in all the literature. In this slide represent the typical GCMS volatile for the Cantaloupe volatile. And generally we are identified around more than 150 compounds identified in compound. And you can see here the compound is in orange color are the carotenoid derived compounds such as geranyl acetone, beta arenol epoxide, phenyl acetones. And while the compound highlighted in the green, they are the aldehyde, and they are the principal volatile of cucumber taste, like 2-nonenol. These are the compounds which are aldehyde. So after analysis, when we already explained you, we identify more than 160 volatile compound. So to represent easily, we divide into 7 categories like ester, aldehyde, ketone, alcohol, hydrocarbon, sulfur, and acids. So if you see that ester is the major contribution of the volatiles, and in the figure you can see here TH9 and TH10 from the California location has a high ester content, and the important ester we identified is the ethyl butanoate, 2-methyl butanoate, ethyl hexanoate, and ethyl benzoate. And these esters are responsible for the fruity odor in the melon. And when we move to the alcohol also, the fruit harvested, alcohol have we found almost in all fruit, and highest concentration found in TH11 from the Texas location and TH13 from the California locations. Similarly, aldehyde, if we move to aldehyde, TH5 generally does not show any significant difference in aldehyde. And aldehyde has fresh, greener aroma, taste, and flavor. And the most common example is 2-nonenol, which has an impact melon-like odor, and which they sometimes give a fresh cucumber taste. So in TH10, you can see that they high in a Texas location, and TH11 they high in a California location. Likewise, we move to the ketone, such as geranyl acetone, beta ionone, which are the byproduct of the beta carotene, but they have higher concentration and they have a floral aroma contribution to the Cantaloupe. And ketones are high in a TH6, you can see here, and TH9 and TH10 variety hybrids. Similarly, hydrocarbons such as limonene and eucalyptol were present, and they have basically the strong antimicrobial property. And if you can see the TH10 from the California and TH13 from the Texas locations, they have a high in hydrocarbon and like the sulfur content. If you look for the sulfur, TH5 from the Indiana, they had the highest concentration in this melons. And if you move to the acid section, you can see that TH5 fruit from the Indiana, and while the acid TH6 and TH1 have a high concentration of acids. So this is basically the overall. We try to we have a large set of data, so we try to understand how each variety with respect to all the phytochemical they work. So here we plotted the PCF of all the hybrid with all the volatiles and all the phytochemicals, including sugar, amino acid, carotenoids, and volatiles. But we try to break down into several formats so we can explain better way. So you can see here is this this is a PC represent the effect of variation with respect to the Brix concentrations. If you look here, the Brix are very close to the y-axis and located in the upper part of the first quadrant. And Brix is the driving component of PC2. And so you can see the Brix are higher in Arizona, TH5, Texas location also, 14, TH1, TH12, and Texas locations, while similarly the lower concentration of Brix are observed in Indiana fruit. So this is spotted by the TSS, where we look at all the melon which we harvested from the Indiana fruit has less than 8 Brix, while lot of the fruit harvested from the Texas, Arizona have a Brix more than 12. So if we move forward, similarly the same trend shown by the carotenoids, the fruits like TH6 from the Texas, Arizona TH5, 14, and TH11, TH12, they have a high content of carotenoids again as compared to the Indiana fruits. So if we move to the total sugar content, so total sugar contents also are close to you can see the sample harvested from the California TH1 here and TH5, they have a good total sugar as compared to the TH9 from the Arizona. And likewise, you can see sucrose concentrations high in the Texas, Arizona, and TH12 variety of sucrose, and they are lower in the Indiana sections. This is also supported by the result we observed that the total sugar content higher in the Texas location, Arizona location. So similarly, if we look at this amino, how all the hybrid behave as compared to the amino acid. So you can see that TH5 and TH1 from Arizona had high glutamine and the GABA concentrations as compared to the hybrid we observe in this quadrant. And hybrid from the California, they have a high in aspartic acid, alanine, valine, and proline, and hydroxyproline. So this is a volatile. So volatile compound, you can see the esters such as ethyl, which mentioned the red color, ethyl benzoate, ethyl 2-methyl benzoate, ethyl hexanoate. They are very closely associated with the TH5, TH11, TH10 from the California fruit. And the other variety, you can see that this is we highlighted in orange. They are the 2-nonenal, geranyl acetone, beta-aronoin. They have a cucumber taste. They impact which impact the fresh cream flavor to melon, and closely related to the TH6 and TH9 from Arizona and Texas TH9 variety. And if we look for sulfur compound like ethyl methyl, you can say ethyl methyl thiopropionate is you can see that they are widely associated with the California TH1 variety, and similarly the antimicrobial compound D-limonene we observed in TH10 and TH6, and lower concentration was observed in this section. So overall, you can see the summary is like the breaks was high in TH5, TH6, and TH9, TH13 from the Arizona, Texas, and California. All the location has a higher Brix content, but except the Indiana. And if we move to the carotenoids, again the TH5 and TH6 and TH9 performing good in the California, in Arizona, as well as in Texas locations. And when we move to the volatile section also shows the TH6 especially and TH12 from the Arizona, and TH9, TH5, TH12 from Texas location has a high volatile concentrations. And in case of amino acid, the glutamine and GABA was high in TH5, TH6, and TH5, TH1, and Texas location from the TH1, TH6. So overall, we observed that with TH5 and TH6 and TH13 and TH12 are performing well in almost on all the location except the Indiana. So this present result output will encourage melon breeding, facilitate the selection of genetic resources for the developing a new cultivar, especially for the region-specific cultivar. So we can get a melon with a high profile and high taste and high Brix content and high phytochemical compositions, which have a health benefits. So moving forward, we also outreach to the different people. So we organized the ACM symposium and in 2021, and we also published a book, Melon Breeding and Genetics, Developing in Food and Quality and Safety. And here we outreach to the scientists, breeder, and student across the world, and we presented our work and present the health benefits and safety and on production of the and genetic improvement of the melons. And again, we also, as I explained by earlier, we are stakeholder field meeting organized in Arizona in the first year, and you can see that here people are meeting and breeding projects and field day. And just a thank you for the group and which helped to facilitate this work, and acknowledgement for the NIFA SCRI grant. And question, thank you, please.
From Jorge Cisneros, how many samples per location are combined to get the measurements presented on the phytochemical graphs?
Okay, so generally we received 5 to 7 fruit from the each location, and we selected minimum 5 fruit for the analysis. So one variety, suppose we are taking a TH5, so we take minimum 5 fruits, and then we make replication from there. We take then we analyze from there. So minimum 5 fruit per sample, per hybrid basically.
So Dr. Singh, I have one question for you. I know you did talk about a number of volatile compounds, and I was looking at the different hybrids that were listed for each volatile compound that was high in, like I know I saw TH1, TH5, TH6, TH9, and some others that kept repeating. So based on the profiling you have done so far, are you able to identify one or two or maybe a few hybrids that have all the desirable volatiles from what you have done so far in the last 4 years?
Yeah, generally we volatile generally present in all except that the level will change. So generally we mainly focus now when we got this result, we overall we look the results and we move forward to the TH5, TH6 and TH9, because then we move forward because we growing this fruit variety again, because this variety is generally good showing the good in what you call Brix content and carotenoids content as well as the volatiles. And some of they have you can say the good ester fruit also, ester smell also. So generally fruit have a you can say ethylbutanate and methylbutanoids. They have an ester compound. So basically, we selected basically pH 5, pH 6, and pH 9. They are the good fruits with have a good flavor. Plus, they have bricks because the flavor taste is matter. So flavor and bricks are both a major contribution for the selection of the bricks, the selection of fruit basically.
Okay. Oh, thank you. Thank you. So are you able to, for example, so practically for the growers, are you able to, if they're mainly looking at the volatile profile, will you be able to suggest a few hybrids or varieties from what you've done so far?
Yeah, we telling them this pH 5 and pH 6 and pH 13, and we are moving forward these varieties basically.
Oh, wonderful. Okay.
So Dr. Ravishankar, to answer the question a little bit clearly. So as you see that the complexity of different components we're using here, basically we're using phytochemicals, we're also using the volatiles. And then along with that, we're using your food safety data, which we need to be considering those, plus the sensory. When we concluded that at current stage, Dr. Crosby's come up with 2 hybrids. But eventually, again, as you heard his presentation, one PS6 is really doing very good, except sometimes it's not like the firm enough. So he finds the 13 is firm enough. So I think he's basically planning for 2 hybrids released now. But eventually, once we have your data on the food safety and other sensory data, I think we're hoping to have at least a few more hybrids out of this project. But again, the challenge we have is some of them, like for example, Dr. Singh presented, some of them are good in GABA and glutamine, but the same variety may not be good on some of the other phytochemicals or some of the volatiles. So it's challenging which one to choose. That's why that whole graphic presentation will help. Okay, some of them are great for certain things, but are we really looking for only health benefits? Or are we looking for the taste and flavor? So that's a kind of a big balance we have to make, because finally at the end of the day, consumer eats what they taste good and what is good for flavor. They don't really worry about the health benefits if you want to sell the melons, but health benefits could be an added one. How do we add that value to them is a good thing. So for example, if we reduce some of these cucumber related volatiles, that's what we need to do. So that's a little bit challenging. That's why I challenged Dr. Singh also we have to come up with some good, all the 4-year data. The good news is most of the work we have done is 4 or 5 years. So I think we'll have some good outcome. At least now we have 2-3 hybrids coming in the pipeline soon. Yeah. But there are a few more once we have all the data combined.
Okay. Yeah, no, this is a great set of data. I mean, it's a lot of data. And to
present
within 20 minutes is not easy. I know. And you've done some sensory work too, I believe, right? And you have some results.
Yeah. So what happens is Dr. Rhonda Miller is really and she has shared some of the information, but because her husband is in kind of in health issues, so she's not able to focus right now for the time being. So once you have her attention, we'll include. We've included some of the data here, but Dr. Singh wanted the raw data so that we can run some analysis here. And again, after that, we'll also be needing your data.
Yeah, thank you. Yeah, thank you so much. Yeah, this is great data. Very nice to see all that data. And all these nice graphs. Yeah, thank you. Now we are going to be doing a demo of how to cut a cantaloupe safely. So Richard has focused the camera in our laboratory kitchen, and I'm going to be starting. So the first thing you want to do is wash your hands with the soap and hot water. So I have washed my hands. My hands are clean, and I am handling the cantaloupe. As you can see, it's right in here. So I'm going to be washing the cantaloupe in hot water with a lot of agitation, with vigorous agitation, as you can see in here. And specifically, you want to pay attention to the area in the stem scar, and also any soil, anything that you see, try to rub it vigorously. And obviously we don't want to be using any soap or anything like that. You don't want anything on your food products. So just, I'm washing it in hot water really well. Lot of agitation again and scrubbing as much as possible with the hand. And the reason you want to handle this carefully is because, as you can see, the melon rind, this is a cantaloupe, is a pretty rough surface. And this rough surface can allow the attachment of microorganisms, foodborne pathogens specifically, such as salmonella as well as listeria. So they can hide in the hard, surface, cracks and crevices that could be in there, and they can, escape treatment. So, as much as possible, you can wash it, and they might still be in there. So cutting the way you want to cut the cantaloupe, you have to handle it really, really carefully. So what I usually do is, and this is what I practice at home too, I would keep 2 separate cutting boards. So, red for contaminated and green for, clean, non-contaminated type of a surface is what I'm going to put. And these have already been cleaned and sanitized, both the cutting boards and both the knives as well. So what I'm going to be doing is, I am going to be making my first cut. And, as a food microbiologist, to me, anything, when I see a cantaloupe, I think of salmonella as well as listeria. That's what I'm thinking of. So I want to handle it really carefully. So assume that this is contaminated. So what I do is, this is again, a very clean knife. It's been sanitized already. I'm going to be making my very first cut. So here are the 2 parts of the cantaloupe. And now that my knife has touched the rind, and research in my lab has shown that if you have any contamination on the rind, the contamination can go into the pulp. Using biophotonic imaging, we have shown that. So now I consider my knife to be contaminated because it has touched the rind already. So I will be washing this in hot water as well as in soap. So we already have soap in here. So with mechanical scrubbing, I'm going to be washing the knife so that I want to make sure I remove the contamination. And we have shown, with research in our lab that, this kind of washing will remove the contamination. So now I have these 2, and I'm going to take, one of the halves of the cantaloupe, and I'm making another slice right there. And now again, the knife has touched the rind. So I consider this to be contaminated again. And so there is one more wash with hot water as well as soap and mechanical scrubbing. So now this knife is pretty clean. So I'm going to be placing this in here. And because I like to work with a smaller knife, I'm taking the cantaloupe in here, and I am removing the rind very carefully and not trying to touch the pulp as much as possible. So the knife is basically handling the rind here. So all the rind has been removed, which I'm going to keep in the contaminated cutting board, which is the red cutting board. And now, since all the rind has been removed, I'm going to be washing both the knife and the cantaloupe again in hot water. And specifically with the knife, I will be using again soap with mechanical scrubbing, not the cantaloupe. So the knife has been cleaned now, and I'm going to be washing the area where the rind was completely removed. So I washed that really well in hot water again. So now that this part of the cantaloupe has been, cleaned, this will not be touching this cutting board anymore. So I place this on the clean cutting board. Now I consider this, as I have removed all the contamination. Same way, you could do it with the rest of the cantaloupe as well. So I'm going to just show you one more piece, one more quarter of the cantaloupe where I'm going to be removing the rind. And the rind should be kept only in this cutting board, no cross-contamination with the green or the clean cutting board so far. So same way, now all the rind has been removed, and I'm going to be washing the knife first with soap and hot water with mechanical scrubbing. Then my cantaloupes are also cleaned here. The area where the rind was removed is cleaned in hot water again here. And so now, because both are clean, these are going to be placed in the clean cutting board. So now we have everything, and same way, similarly, I would be doing another slice, washing the knife, then again, removing all the rind. And once all the 4 quarters have been put in there, I'm going to be just quickly washing my hand one more time. I know it's a lot of washing, but it ensures food safety. So now I have washed my hand, and I'm going to be now handling the clean cutting board. So I'm going to set this aside. The clean cutting board is going to be handled, and both the knives and the cutting board are clean. So now what I will do is, with the clean hands, I'm going to be slicing my melon, and this can be stored in the utensils, and it can be refrigerated right away, or it can be consumed with some peace of mind that, food safety wise, it's going to be much safer than, handling in the way you want without washing. So this is one way of safely cutting the cantaloupe. So, the thing here is, it's better to have 2 separate cutting boards, better to, wash your knife after each time it touches the rind, and then, keeping them separate, the unclean and the, clean portion of the cantaloupe. So, and this can be stored and it can be consumed. So this is, this is the way I do it at home. Does anybody have any questions? Oh, okay. What about turning the faucet off with contaminated hands? You'll have to, you can clean the faucet, and before you handle your clean cantaloupe, that's why I washed my hands before handling the clean cantaloupe. It's really hard to avoid that, but with the even though you're turning it on, then you're still washing your hand with kind of soap, and you're cleaning your knife with soap as well. So hopefully that takes care of it. That's why, like, once I had all the clean cantaloupe on my clean green cutting board, I went ahead and washed my hands again, and then I sliced the cantaloupes with a clean knife. Hope that answers your question. Is the sponge contaminated after washing the knife? That's a very, very good question. So the best way to deal with the sponge, after you have cut a cantaloupe, would be to take the sponge, in a container. You try to wash the sponge as much as possible, but then you can, put it in a container with water, and you can microwave it for like 6 minutes. Or the other way would be to, boil water in a container, and in the boiling water, you may want to, just treat the sponge in the boiling water, probably for like 5-6 minutes, and then reuse it. Because in my opinion, as a food microbiologist, if I believe, the rind and the knife are contaminated, the sponge can be contaminated, and sponge can, absorb bacteria. So that would be the best way to, decontaminate the sponge. Now we will go ahead and begin with the food safety presentations. So the first presentation, so far what we saw is how the melons are grown, breeding. We looked at the analysis of the volatiles, and now we are going to move on to the food safety part. So the first presentation will be looking at the prevalence of foodborne pathogens and indicator microorganisms in melons that were grown in Arizona as well as in California. The presenters are Richard Park and Madison Goforth. Richard Park is a research technician in my lab. He has been working, in my lab since 2017 or 2018. He was a student in my class, and then he joined the lab, and he's been one of the leads in the Melons project. He's been handling a lot of the work in the Melons project, and he was even the organizer and the coordinator of this workshop. I would say he took a lot of effort in organizing this workshop. So thank you, Richard. So Richard will talk about the prevalence of pathogens and indicator microorganisms from the melons grown in Arizona. And Madison Goforth is a graduate student in Dr. Kerry Cooper's lab, and she has been one of the students working in the Melons project for quite some time. And Madison would talk about the prevalence of pathogens and indicator microorganisms from the melons that were grown in California. So Richard will go first, and he'll be followed by Madison.
Okay, thank you everyone for attending this workshop on such short notice. So the thing that I will be covering in this PowerPoint is the prevalence of foodborne pathogens and indicator microorganisms on Arizona grown commercial cantaloupe melons. Madison will cover California and the environmental samples. So basically the objective of this study was to isolate any foodborne pathogens and to quantify the indicator microorganisms from the melons grown in Arizona. And the overview of this project was to analyze over 1,000 melons for any of the prevalence of the foodborne pathogens, looking at those indicator microorganisms. So this was over the course of 4 years, as you know. In year 1, we sampled 228 cantaloupes. Year 2, we tried to bump that number up just so that we could fit the 1,000 within 4-year time frame and not give too much work in the later years. So we did 339. However, in year 3, of course, COVID-19 was rampant throughout the nation. So we had to not sample any prevalent melons. So basically all those leftover melons we had to do in year 4. And we did 435 cantaloupe samples and the environmental samples from each of the fields. So in terms of how we collected these environmental samples, we did soil, root, and air samples. So the root, Martin collected the root samples after the commercial growers harvested the cantaloupes. He went to their fields, pulled the roots up. Soil samples, he collected in a Z pattern, as you see in the middle, bottom middle here. Just to add that randomness to the sampling procedure. Air samples, we use sedimentation plates, and water samples were collected through a modified Morse swab. You can see this cheesecloth on the bottom right here, just helps collect the water that it's pumped through. We pumped 10 liters through this container that holds the cheesecloth through so that we can get all those particles and microorganisms in the water sample. So I know there's a lot to look here, but basically all these on the top are the samples, whether they're the melon samples or the environmental samples. Basically, we took those samples. For melons, we created composites of 3 just to make the work easier. So 3 cantaloupes is 1 composite, and we enriched them. We afterwards, we enriched them. We sort of massaged them to detach those microorganisms there. And we collected some aliquots for the indicator microorganisms. So for indicator microorganisms, we diluted it in PBS, and we plated it on 2 types of media, ECC, or E. coli coliform agar, and M. enterococcus agar. So on the top right, and basically everywhere but the bottom middle, is ECC agar. You can see the purple here on this type of agar. Purple indicates coliform. So we counted the coliforms. Here you can see on the top right, a blue colony or a blue-green colony. That's indicative of E. coli. So we collected those E. coli samples on any of these plates that we collected for further analysis. On the bottom right, we saw these red colonies. We did further identification to determine what that was because that wasn't part of the color schematic that was given to us. And here on the bottom, these red colonies on this plate is Enterococcus. So red indicates that it's an Enterococcus species. But going back to our samples, we further enriched them and inoculated positive controls so that we know our conditions were correct in incubating and growing Salmonella and Listeria species. Afterwards, we did our double enrichments either for Salmonella or Listeria. Salmonella went through a double enrichment of tetrathionate broth here, further through M broth, and then streaked on XLT4, which is a selective and differential medium for Salmonella. For Listeria, it was a little bit easier. We just had to do it in LEB, so you can see all the bottles of LEB. And after they're enriched, and we plated on rapid L mono for Listeria monocytogenes. We also took some aliquots for glycerol stock in the -80, just in case we did get a presumptive positive, we could go back and do further analysis. So what did we see on our XLT4? We saw these black colonies. On the bottom, you can see Salmonella positive. They sort of look like the top, but not quite. You can see there's sort of sparsed out. What we did was we isolated these black colored colonies. We found that they were kind of goopy. They were very thick, as you can see, not very clean. After increasing the incubation time, we actually saw that the blackening started to occur. We did perform a gram stain. We saw that they were gram-negative raw. So we couldn't really differentiate if it was Salmonella or not. So we did an API 20E on the samples, and we found that those goopy colonies were actually Rallutella planticola. We also plated those ECC chrome augurs, those red colonies, and we found that a good identification was Serratia rubidea. So luckily it was not Salmonella. So in terms of the data we'll display, just for the essence of saving time, we'll just look at the first 2 years. Here we can see that in terms of the indicator microorganism counts, we do see high indicator microorganisms in these melon composite for both Enterococcus and coliform counts. We do see it high for soil as well and the rhizosphere root samples, though for water and air, they tend to be lower. For year 2, we did again find that those melon composites were high, as well as the soil, the rhizosphere, apart from sometimes the air did collect a lot of indicator microorganisms, but overall not much, and the water was lower as well. And over the course of the 4 years we performed this project, we did not find any positive or presumptive positive strains of Listeria or Salmonella species. So to summarize our results for Arizona specifically, we didn't find any Salmonella or Listeria in either the melon composites or the environmental samples. Air samples had the lowest level indicator microorganisms. Basically, again, we did the sedimentation plate, leaving those TSA and non-selective auger media out in the field. Martin would leave them for 30 minutes to just collect those samples, the air. And for year 1, again, melon samples did have that highest level of indicator microorganisms. Year 2, root samples had the highest, along with the melon samples. All fields that we collected, or Martin collected, thankfully, these cantaloupes had signs of wildlife, bird droppings, nests, visible animals, such as birds specifically, more commonly, animal tracks, and rodent tracks. 2 melons were positive for E. coli in year 1, and 1 melon sample for year 2. After further analysis and further tests, we didn't determine any pathogenic E. coli in those samples, luckily. And we have to remember that, again, Arizona melons is a field pack state. So the melons here are unwashed, they're harvested from the field and brought to the consumer for purchase. And we also found, again, that melon root and soil had higher levels of background than air and water. Air samples obviously had the lowest because we were doing sedimentation plates and not a sampler like an Anderson sampler or something like that. So thank you. And then we'll take questions after Madison.
Hi, everyone. Like everyone stated, my name is Madison Goforth. I'm a graduate student in Dr. Kerry Cooper's lab, and I'm just here to present on the prevalence for California samples. So the 2 sample sites, we did Northern California as well as Southern California. We took composite samples of air, soil, reservoir samples, as well as melons. There was no irrigation water samples available, but we also did melon microbiome. So for Northern California data, what we've looked at was for E. coli, there weren't any presence of the strain, but there were coliform counts between certain percentages. Some range between 0 for certain soil samples, while some were above the normal count for reservoir samples as well as melon samples. For salmonella, we did a cocktail mixture of enterica, Newport, and Madison. And what we found for our air samples, composited, by the way, were 3 of 5 were positive. We did do PCR testing to confirm that they were these, as well as we did controls. Lastly, we did also a Listeria cocktail mixture of monocytogenes, 101M, F6854, and Scott A. And same thing for the air, 2 of the 5 were positive. And samples were shipped for the PCR verification, and those 2 were confirmed to be positive. Lastly, for melons on salmonella cocktail mixture, 8 of the 84 were positive with PCR verification. Originally, we had done 10 based off of the like qualitative methods, and then we shipped them off for quantitative. For Southern California, we didn't see any E. coli, salmonella, cocktail mixture, or listeria on any of our air, soil, rhizosphere, or melon samples. However, there were the controls were positive. Lastly, to summarize, upon PCR confirmation, Northern California showed more prevalence for foodborne pathogens in air composite samples, as well as some melon composite samples, and some future outlooks could be possible regulations of field operations and staff that could affect results besides the regionality where they're from. Further collection and higher sample collection points may be reliable in tracking sources of fecal matter and foodborne pathogens. Thank you. And here's some acknowledgements.
So thank you, everyone. Are there any questions for Richard as well as Madison? I see one question here. So Richard, you want to take it?
So one of the questions is about the coliforms or the normal flora on fresh fruits and vegetables. What do you think they're indicating on Arizona melons? So we saw that there are higher numbers. So what the indicator coliform counts are looking at is some of them can be the relations to pathogens. So for indicator microorganisms, the higher the number, the higher the chance of foodborne pathogens being present. What the exact ratio is a good question to be further studied. But we do know that the higher the prevalence for those indicator microorganisms, the higher the chance of a foodborne outbreak occurring. And which is why that in microbiology, they use indicator microorganisms because one, they have to be present even regardless of the pathogen being there, because there'd be no point to have an indicator microorganism that could disappear, but Salmonella or Listeria being present. It has to be in high enough numbers that supersede or exceed the foodborne pathogen. Otherwise, you would be easier to grow the pathogen itself. And also that they have to persist throughout. So to say that these indicators are meaning that for sure that there's going to be a foodborne pathogen on the melon is not correct, because those indicator microorganisms are just indicators. They're just giving us sort of guidelines or warning signs, how clean these melons, how clean the environmental samples are. So I hope that answers your question. So Natalie Brassel said, commented just to mention too, that generic E. coli is the fecal indicator organism, not total coliforms. Total coliforms are always on soils. So thank you for that. And yes, thank you. Yeah, luckily there's no Listeria or Salmonella in Arizona grown cantaloupes, especially since these are the commercial ones we were testing.
So thank you, Richard, for the wonderful presentation. So now that we have seen, what the prevalence of pathogens and indicator microorganisms are on Arizona and California grown melons. Next, we are going to look at again field level. So again, we are trying to go from farm to fork. So we are going to next look at, again, Richard is going to be presenting, and he's going to talk to us about what kind of studies he did to look at the cross-contamination potential of soil and dust onto different melon varieties that we used in this project. So Richard, it's all yours. I already introduced you.
Yes.
So,
No worries. So now this research, we're looking at the cross-contamination. So basically from the environmental matrices we're looking at is soil and dust. How much of those foodborne pathogens, specifically Salmonella and Listeria, will transfer over from soil or dust onto the melon rind itself. So let me just, I think, it's pinned, but just move on. So the reason why this research is important is because there's been multiple outbreaks. For Salmonella enterica since 1990, there's been over 14 outbreaks linked to cantaloupe or just melons in general. In fact, in 1990 to 1991, there was an FDA survey of imported cantaloupes and watermelons specifically from Mexico. And during that sampling procedure, they actually found that approximately 1% of the rind was contaminated with various Salmonella cerevars. So that was kind of alarming. And in 2018, there was an outbreak of Salmonella Adelaide with around 77 cases, only 36 hospitalizations, but luckily no deaths. And then the following year in 2019, an outbreak of Salmonella Caro with 137 cases, which resulted in 38 hospitalizations. But luckily, again, no deaths, and these are pre-cut melons. So for Listeria monocytogenes, on the other hand, most of us remember the infamous 2011 outbreak of Jensen Farms, which there was 147 cases of those. 143 of them had to be hospitalized. 33 of them, unfortunately, passed away, and there was one miscarriage. So you can see amongst the two pathogens, Listeria monocytogenes is the more dangerous pathogen. However, Salmonella, as we see through the amount of outbreaks, is more common. So basically, again, the objective of this study was to investigate soil and dust as vehicles of cross-contamination of foodborne pathogenic bacteria to the melon crop itself. So we had to do some preliminary work. We had to determine whether stomaching versus sonicating. So stomaching, as you see here in the bottom center picture, simulates peristalsis of the intestines, even the stomach. These two paddle blades will beat back and forth, helping us detach the bacteria on the melon rind itself. However, versus sonicating, which on the bottom right, we see an image of, it uses sound energy to shear the attachment strength or this attachment of the bacteria. But for sonicating, there's a fine line before you lyse or kill the bacteria cell and detaching it. So we had to determine which was the best method. And in terms of this research, because of that dirt, sonicating was better because if we use the stomacher, those dirt particles can rip open the stomacher bag and all the contents can leak out. We also had to determine how we wanted to create the dust and how to inoculate in the pathogen. So how we came up to inoculate the melon rinds with dust was to create this box, which you'll see later, and basically spray the dust onto the melon rind itself using a pressurized air can. We also had to create dual antibiotic resistant strain of Listeria. As you see on the top right, it's completely covered in background growth that we can't determine which of those colonies is Listeria. So thanks to Chi, the visiting scholar, he was able to create a dual antibiotic resistant Listeria strain. As you see in the lower picture, we have only Listeria growing on this auger. And with the help of many undergraduate students, we had to have hundreds and hundreds of plates and liters of broth. So in terms of exactly how we prepared soil, basically through two different sieves, a number 20 for soil, and then a double sieve of a 20 and 100 to get that dust. So we can see that obviously the soil sample is a lot more larger than the dust samples. We also, after inoculating, had to re-crush those clumps of dirt back into fine dust powder. So this is the before and after inoculation. Otherwise, this doesn't really simulate the dust environment that we're trying to simulate. So of course, when melons arrived to Tucson from other states, we had to expect the worst. So as you can see here, the boxes fell apart. There's too many boxes stacked on top that it just wasn't structurally integral, and they just fall onto the ground. They'll get lost in transport. One of the first years, the very first shipment from Georgia was lost for a while. And we just had to go through them and inspect them for any mold, cracks, discoloration, bruising. We had to dry any melons so that wouldn't have too much condensation, creating mold rooms in our cold rooms. We had to count the number of usable melons because we had to see, did we have enough to do certain experiments? How many experiments could we do? And of course, we had to separate overripe melons from ripe melons so we know which ones to prioritize before they go bad. So in terms of the research work for Salmonella and Listeria, we used Salmonella enterica cerevar Newport, which causes around 100,000 cases in the US annually of foodborne illness. Ours was resistant to ampicillin and streptomycin. For Listeria, we used the strain F6854, which again, through cheese help, we made it resistant to rifampicin and streptomycin. We tried with ampicillin because that way would make it easier in terms of the antibiotic use to keep it simple and not have to work with rifampicin. But it was just too difficult and the time did not allow for that. So in terms of the procedure, we cut the flesh or the edible part of the melon. We just wanted the rind and the thin layer afterwards. So we cut them into 10-gram rind samples, and we washed it through double tap water, double DI water, after which we dried, and we used ultraviolet light for 15 minutes on each side. So we either did the dust or the soil experiment. So the dust, we just inoculated dust with, or soil with the Salmonella or Listeria, and we placed the rind samples on the soil sample and we put weights there. It's 100 or 1 kilogram, which is a very, very light melon. But the most important part is that these weights help have the melon rind come in complete contact with the soil. If you look on the right, you can see how the melon rind actually curves upwards. So you want to get that full contact with the soil. And we had a contact time of 1 hour before we started collecting the samples. For dust, on the other hand, we inoculated only 3 grams of dust. We didn't need too much. We used a dust transfer box, which is just a simple sterilite box. We have the dust sample here in a bifurcated centrifuge tube just to collect it in one spot. So if we moved it, it wouldn't move across the entire box. And we aseptically taped the rind onto the other side. We used a pressurized air canister, and with like half a second spurts, only one, we sprayed the dust in an enclosed container. So afterwards, we collected the environmental samples, 1 gram of dust or 1 gram of soil, just to see the source of contamination for determining the percent transfer. After the contact time, we cut the rind and resuspended it in phosphate buffered saline, where we sonicated it for 2 minutes. So here we have Salmonella Newport at a sonication setting of 40-40, Listeria is 40-60. What those numbers represent is the duty cycle and the output cycle. The duty cycle, meaning how long is the sonicator on for when it shoots out those sound energy waves. The output cycle is how powerful. So both of them have the same duty cycle, around 40%, but the output, you can see that Listeria monocytogenes required a little more strength to remove that. Afterwards, we centrifuged the sample. So when we sonicated, it didn't look like there was any dust collected or any dirt on the rinds itself. However, after we removed the rinds and we actually centrifuged the suspension, we actually see there's quite a bit of collection of dirt or dust in there. So to us, to the naked eye, we couldn't see it, but after centrifuging it, there's definitely some residue there and some transfer. And then we removed the supernatant, we resuspended it, and we plated it on XLD for Salmonella and MOX for Listeria. So what do our results show? For one of the years, this is the results being displayed. We do see TH7, one of the hybrid cantaloupe netted melon varieties, having the highest percent transfer here, while a HD 150, a honeydew variety, had the lowest percent transfer of only 0.0015%. While for dust, we do see very significant lower numbers somewhat, the highest being for Infinite Gold Cicada. It is a cantaloupe variety with that netting again, with 0.0108%. However, we do see significantly less percent transfer with Da Vinci Cicada, F39. Those are the netted melons, and OC164, which is a smooth honeydew type melon. And we only saw a percent transfer of 0.0004001%. Basically, how we calculated percent transfer was take the population of bacteria on the destination, which was the melon rind, and we divided that by the total population of bacteria on the melon rind and in the environmental sample, and then obviously multiplied by 100 to get that percent. For Listeria monocytogenes, on the other hand, we did see again TH7 having 0.1112% transfer, while HD 150 having 0.00305%. So we do see again that the honeydew or the smooth melon, including HD 252, another honeydew variety, having low percent transfer. For dust again, it's significantly lower than we see with the soil numbers, with F39 at 0.00679, and surprisingly an Infinite Gold Cicada with only 0.00002%, so 4 zeros. So surprisingly, what we saw is in the first year with Salmonella, we saw that TH2 was the only one where soil had lower percent transfer than dust. So it was the only outlier. While the rest, soil percent transfer of the foodborne pathogen Salmonella and Listeria was always higher by a significant degree of significant difference of less than 0.05 on soil than dust. So to summarize our results, again, the percent transfer, like I said, is significantly higher in soil than dust, but we expect that because again, the cantaloupes are in full contact with the soil for an hour. While dust it's being sprayed, it has to stay into it has to get into the melon grooves, into the netting, or just settle on it to collect. So again, we saw 13 out of the 14 for Salmonella Newport, and all of them for Listeria monocytogenes. The percent transfer was higher for Salmonella than Listeria on 11 out of the 15 samples. So we saw that there was higher percent rate of Salmonella than Listeria, possibly due to just Salmonella being more hardy in the environment. So it could just survive that 1 hour contact time versus Listeria. And we only saw that very few cultivar and melons actually showed significant difference for percent transfer between dust pathogens, meaning looking at all the dust percent transfers, were there any melon cultivars that were significantly different? So only 6 out of 15 of them actually showed significant difference from each other. And in terms, we had the highest percent transfer for each state, such as for soil, we see TH7, Da Vinci Cicada, OC164 for Indiana. So most of these, apart from OC164, is a netted melon, that one being more of a honeydew. Dust, we saw F39 and Infinite Gold Cicada, again, 2 of the netted melons. While for Listeria, we do see quite a few with similar numbers, TH7 showing greater than F39, TH4 greater than TH2, and Infinite Gold Cicada. Again, all those are netted melons showing high percent transfer rates, while dust was F39, a netted melon, and HD 252 for Indiana, a honeydew smooth melon with no netting, really. So we did see that very few netted versus non-melon showed significant difference, meaning was there a significant difference between having a cantaloupe, having a melon with a netting pattern versus a non-netting pattern? For in this instance, we only saw that for Salmonella, only 1 out of the 3 hybrids and 3 out of the 6 cantaloupes actually showed significant difference from their smooth counterparts that was grown across Arizona and the other states. For Salmonella Newport, for dust, none of the hybrids really showed significant difference between netted versus non-netted, again, because of how dust has to get trapped into the netting. and only 1 out of the 6 of the cantaloupes, that being infinite gold cicada. For Listeria monocytogenes, on the other hand, we did see all the hybrids and half of the cantaloupes that we tested showing significant difference, that netting versus non-netting, that there's a higher percent transfer. For dust, again, there wasn't much difference. Again, none of the hybrids and only 1 of the cantaloupes actually showed significant difference between netted versus non-netting in terms of percent transfer. So in conclusion, we know that melons are grown on the ground, so they can be contaminated on the surface with dirt, chemicals, animal excrement, and or bacteria. So we have to be wary of that. Soil has the higher percent transfer than dust. We saw that 27 out of the 28 varieties, soil had higher percent transfers of both Salmonella and Listeria than dust. And again, like I mentioned before, Arizona melons are field packed, unwashed, there's dirt on there. And if that dirt happens to have foodborne pathogens, the consumer or the processing plant that is processing those melons are at risk. So growers have to be mindful of dust as well. We did see that it was very low in terms of percent transfer, but it wasn't 0%. So dust is a source of potential contamination. And also processors and consumers have to be wary of debris on the melon surface as well. So from this study, we could understand the risk of environmental matrices like soil and dust, of the potential it could for cross-contamination from onto melons from field conditions. And this data that we are collected over the past 4 years can be used for a science-based risk analysis and also allow us to create appropriate control measures to prevent these risk factors and also determine the risk that the consumer is exposed to foodborne pathogens. And also if we need to create any new prevention or control measures that are needed by the industry. So I'd like to thank everyone for this presentation. So is there any questions?
So the number of samples in all of these is a small number, right? Or am I wrong?
The question was, for this research study, was this sample number small? So in terms of how many replicates we did, it was dependent on the number of varieties. So if we go back,
Right.
you can see the different varieties here. We had 8 for Arizona, 4 for California, 3 for Indiana, and 1 for Texas that we sampled for the study. Each one of these, we did 3 replicate tests, and we did double plating to make sure we plated duplicately on the XLD or modified Oxford auger to make sure that our numbers were consistent. And so we went through the basic standard that is universally accepted of the number of replicates for this study.
So with the data that you got from this, what would be the next step if you found, well, you found some changes that looked significant, and then if you were going to do this again, like TH7 on
Yes.
Arizona, how many individual melons would you use in the next level of trying to find out if in fact it has a transfer of the salmonella?
That's an excellent question. So the question was, what are the next steps from this data that we collected, such as from TH7 with the high transfer rate? So this is a multi-university project. And like Dr. Crosby said, we're going to try to correlate and compare the data that we collected here in our food safety testing to their melons that they grew and see which ones, like such as I believe he said TH6 that he was putting out hopefully to the market and releasing it soon. From our data that we collected for TH6, what is the food safety data? And how does it compare to the commercially grown varieties, such as Da Vinci Cicada, F39, those. For future work, we can also look at, depending on, is there a true difference in the netting density? So we'd have to simulate, we could simulate netting patterns and see how much of that impacts the percent transfer rate. And also looking at the chemicals that these melons produce on the surface, can those have any antimicrobial effects that will prevent either attachment or prevent growth of the pathogens itself?
Does that answer your question?
Yeah, I'm just trying to get an idea of where this is at. So there are different universities who are doing the same thing today with the same kind of pizza and a dog in the lab. And there are a few at this level working on this. And then from what you find, then the next step will be to say, all the different colleges found an issue with, again, I'll just use TH7. Then it would be, oh, let's go further with this. And then instead of the sample size of basically a handful of melons, you would start using hundreds of them. And that way you can then more finely understand what the correlation is. What the correlation is on the excessive transfer. Because then you can even go further and look at what topological aspects of the melon relate to that.
So your question, Marcus, with the number of melons that you have sampled, right? So, yeah, I mean, with this research, I think all the hybrids were cultivated at least for a couple of years, 2 to 3 years. So each time we got the same cultivar, we repeated the experiments. So I would say some of the THs were done, from, if you remember from Martin's presentation, some of the THs were done in multiple years. So each year, the same cultivar was evaluated. But here he's just, Richard is just showing some samples because it's a lot of data.
Yeah.
So he's just giving you results of, some sample studies, not the whole thing. So, I mean, but again, because these are special cultivars and they can be grown only to a certain number, we could only get a few number of melons. But
of course,
as you say, if we find a correlation, then it is definitely, between food safety and, volatiles, disease resistance, all the various, parts of the pieces of the puzzle, then, as you say, yes, it makes more sense to do the same cultivar, maybe, many melons. Yeah, that's possible, definitely. But now, because, they were trying to come up with a cultivar having the desirable attributes, and the study is only 4 years, it all depended on, whenever we had that cultivar planted, we were able to sample it. Yeah, but what you're saying is a great point. Once we do the correlations, we can definitely go for a larger number of the same variety samples.
Thank you. Yeah, I'm trying to get an idea of an image of the whole process that you're doing. And like you're saying, since you're doing the same thing for a number of years, and it's each time it's not a great number of samples, but it's probably enough to look for differences, because if you find differences from what you would expect, then that says, oh, well, maybe there's more here. And as we go further into this, we might find that there's some structural aspect of the netting, for example, that either holds on or resists or lets go.
Yeah.
Thank you.
Absolutely. Yeah, we can definitely do that. Yeah. No problem. Thank you.
Okay, and the next question was from Nitin, who said, why are there too many decimal places while assessing the transfer rates, such as for the dust and soil samples? That's a great question. The reason why we have so many decimal places is because, again, the formula is the population of bacteria on the destination versus the population from both the source, which would be the dust specifically for these tiny numbers, and how much transferred over. So very little bacteria transferred over in terms of dust samples, which is why we had such low numbers. And we inoculated the dust with very high levels of pathogens, which is why we could get those pathogen counts. Otherwise, if we inoculated it with too low of a number, there could be possibly no recovery that we would find in terms of using, in terms of dust being a source of contamination. So I hope that answers your question.
All right. So now that we have seen that these environmental matrices can serve as a source of contamination onto the melons, we are going to move on to the next step, which is we are going to look at how these pathogens, attach. What is the attachment strength of different foodborne pathogens onto different melon varieties that were tested, evaluated in this project. So the next presenter is Kirat Bains, and she is going to talk to us about the attachment strength of two major foodborne pathogens of concern on melons, Salmonella as well as Listeria monocytogenes. Kirat is a graduate student in my lab. She's been in my lab for about an year, and she's been involved in the melons project. And she's also doing some other things. She's also looking at biofilm formation. She's looking at efficacy of antimicrobials against pathogens on different types of surfaces. And this is one of her projects. So she's going to tell us about, what was done and what they found. Okay, so Kirat, it's all yours. Thank you.
So thank you so much, Dr. for the introduction. Well, to introduce myself again, my name is Kirat, and I'm a grad student in Dr. Ravishankar's lab. And so I'm here basically, so I'm a grad student in Dr. Ravishankar's lab, and today I'll be talking about a small project that we did under the Big Melons project. And so our project basically studies the attachment of specific foodborne pathogens on the rind surfaces of different melon varieties and hybrids. So the title of our project, as seen here, is Attachment Strength of Salmonella enterica, cerovar Newport, and Listeria monocytogenes on melon hybrids grown in different regions of the United States. So for this presentation, I'll start this presentation with a brief introduction of the concept on which this experiment is based on. And further, I would talk about the specific objectives of this experiment while also discussing the procedure that we followed to do this experiment. At the end, I would discuss the results and conclusions that we could get from the data obtained from this experiment. So let's go ahead. And so as we know that outbreaks from melons have been an issue of concern recently, and outbreaks due to whole and pre-cut melons have been reported from time to time. And as seen here, there are two examples of such outbreaks, and in previous presentations also, we discussed why we are doing this project and why the food safety aspect of melons is of great importance to us. So one such example of Salmonella blended up outbreak of 2021 ended up infecting more than 400 people. And luckily, there were no deaths in this. But in listeriosis outbreak of 2011 that occurred in US, it ended up infecting more than 140 people. And at the same time, it ended up in more than 30 deaths. With that, the very recent melon outbreak that has been reported was in 2019 from pre-cut melons. And so that ended up infecting more than 90 people. So definitely it is an issue of concern, and we really need to concentrate on working to make it more safe in future. So the sources of contamination in case of melons could be the irrigation water that we are using. It could be the soil or the animal intrusion, and probably the texture of the melon skin could be one of the reasons that is making it more susceptible to these, susceptible to the attachment by these foodborne pathogens, and hence the contamination. So also melons can get contaminated during different steps of harvesting or processing. So all in all, this tells us why we are doing this project and why food safety is important in this case. But our project basically aims at identifying the melon hybrid or the melon variety, which has a lower foodborne pathogen attachment. That is what we are trying to do through this project. So the specific objectives of our project is to evaluate the attachment strength of Salmonella Newport and Listeria monocytogenes on the melon rinds of various melon varieties and hybrids. At the end, we would compare the results obtained between the two pathogens as well as the varieties of melons. So to do this experiment, we washed our melons thoroughly, thrice with tap water and thrice with DI water. And further, melon discs were cut out of these melons with a coat borer. Everything was done in an aseptic condition. And these discs were further treated with UV light for around 15 minutes for each side so that we do not have any bacterial contamination or any inherent bacteria from the melon itself. Further, these discs were inoculated with an overnight culture of Salmonella Newport and Listeria monocytogenes. These discs were allowed to sit for 30 minutes to facilitate the attachment of these foodborne pathogens on the surface of these on these rinds. And then these melon discs were vortexed in a solution of PBS with twine to remove the loosely attached bacteria. The same discs were further sonicated in BPW to remove the strongly attached bacteria. These solutions were further serially diluted and plated to calculate the population of these bacteria. And further, the attachment strength was calculated by dividing the CFU per ml of strongly attached bacteria by CFU of total attached bacteria. So the results, the data obtained from this experiment is as seen here. So this is the attachment strength values that we obtained for Salmonella Newport, Salmonella enterica, Serovar Newport, from the melon hybrids that were grown in 5 different places throughout the United States. They are mentioned here, that is Texas Vislaco, Texas Uvalde, Arizona, Indiana, and California. And we have around 16 different melon hybrids and varieties, which are mentioned on the extreme left of this table. So because it is a very busy slide, we'll discuss the results that we obtained in further graphs. And we have a similar data table for attachment strength values for Listeria monocytogenes for these different varieties and melon hybrids that were grown in, again, different parts of the United States. So this graph, as we see here, gives us the average attachment strength of Salmonella Newport as well as Listeria monocytogenes on various melon hybrids that we obtained. And I think these melon hybrids have been discussed in previous slides as to how they were bred and what was the idea behind it. This shows that TH9 has the highest average attachment strength for Salmonella. And if I go further, so TH12 has the highest attachment strength for Listeria monocytogenes, and TH1 has the lowest average attachment strength for Listeria. And so this is our figure 2, and these are the remaining melon varieties that we received. So here we can see out of all the results, SMA variety of melons showed the lowest attachment strength of Salmonella. So to conclude this, we can say that the attachment strength for Salmonella Newport was lowest in case of SMA melon variety, while it was highest in case of TH9 melon hybrid. Further, for Listeria monocytogenes, the average attachment strength was higher in TH12, while it was lowest in TH1 melon hybrid. And we did not observe any significant difference between the average attachment values for both of the foodborne pathogens that we tested in this experiment. Further, these melon rinds that we tested were sent for SEM analysis just to see if there was any correlation between the roughness of this roughness of the surface with the attachment strength data that we derived from this experiment. And at the end, we can say that definitely we need some effective interventions, because this experiment definitely proves that foodborne pathogens can attach to melon surfaces in different capacities. So definitely some effective interventions can help us get rid of them. So at the end, I would like to thank Dr. Sadhana Ravishankar for giving me this opportunity to present this work in this workshop. And I would like to thank Catherine, David Rollins, Richard Park, Libin Zhu, and Aishwarya Rao for training me to do this work. Thanks to all Ravishankar Lab members and volunteers for their continuous support during this work. And the study was supported by USDA-NIFA SCRI. So these are some of our collaborators, and thank you so much. So I'm open for questions.
So Sadhana, I have a question to the student. It's a good presentation. I really like that. So one question is, if let us say we are planning to release this TH6 now, it looks like the attachment strength is not lowest, but it is lower. What is your recommendation? Can we consider that as a good hybrid to be released and consider that as a food safety resistant variety, you think, based on this attachment only?
Okay. So I think as per the data, because we do have certain hybrids that have a higher attachment strength. So I think we can recommend and we can say that as far as food safety is concerned, and when compared to other hybrids, this is a better hybrid, which we can go further with. So that is what my answer to this would be. But I would, if Dr. R would like to add something.
Yeah, Dr. Patil, yeah, that's a great question. So I think it looks like TH6 came up when I remember, looking at the slides from Dr. Singh with regard to volatiles as well. So we probably definitely, need to look at. So what Kirat presented is from just 1 year worth of data. And
Right.
we do have other years that, also need to be analyzed and so on. So once we have all that analysis done, and once we do like a, correlation of food safety with the, studies in your lab, and if we are able to see the TH6, is better in all aspects, I'm pretty sure we can definitely do that. So
Okay.
but as of now, it looks like, the attachment strength is, not the highest,
Yeah. Okay, yeah, this also provides some clue to the question by the previous audience earlier. I think there was a question about how many times you have to replicate it also.
Absolutely, yeah. One of I know one of the people in the audience, Matt Cook, he did mention replicating, and I think that would be a good thing to do after we look at the correlation, and then maybe just look at those hybrids that we want to do, maybe some more replicates, and like just to increase the number.
Okay, yeah. Thank you for a good presentation.
It seems to me that what you're doing is you're using flooding to remove. Are you doing anything else like air, blowing them with air or anything? So it's either dissolving or mechanically using the water and like the manual work on them to remove the microorganisms. So it seems like the microorganisms aren't terribly strongly attached, but more like they're in crevices and edges and things like that. So the skin or the rind is really pretty tough. Interesting.
So what is your question, Matt? But I mean, the rind in some cases is really, really rough. And that's the reason we had to go for the procedure that, Kirat is using.
Yes.
And again, your point of them hiding in cracks and crevices is extremely valid. That's the reason we have to go for sonication, because, just by, washing, you're not going to be able to remove those. Absolutely.
So when you're processing large numbers of the melons, you could have in the chain of events, you could have some kind of a bath that had sonication in it that may, and even spinning them because of the I'll call it wave motion, almost like in a jacuzzi, and so that could end up being a practical result of your research, because you're showing that it's not necessarily difficult to clean them, and that therefore, when it comes to processing large volumes of not just the melons, but other food stuffs that have similar characteristics, that you can use some aspect of what you were using in the lab enlarged to a much larger size.
Yeah, absolutely. I think what you're saying is a valid point. If you want to remove all these bacteria, and if you're doing like a continuous processing, I think what you're saying makes sense. Have like a large, water tank or something, have this sonication and some kind of agitation, rotation, motion. That way the microorganisms can be. Absolutely, yeah. At the industrial scale, if you're doing this, you probably have to, do that.
And that's what happens with the research that you all are doing, because you're just doing it in almost a minimal manner and coming up with data that allows you to say, this looks significant. And then from there it grows. And then in 5-10 years, there's a Kirat machine on every, I worked with chili processing with designing equipment for it. And it's a similar kind of thing that a lot of what it is, it starts with people having had done research. And then the question comes up, okay, what is the issue here? And the issue is Salmonella, Listeria, E. coli, whatever it is. And then the information, the knowledge just gets shared.
Yeah, absolutely. Yeah. Absolutely. I think
Thank
what
you.
what you're saying makes sense. And that can be even done with hot water immersion.
Yes.
Because,
Yes.
for melons, some people use hot
steam
water
spray.
immersion. Exactly. Steam spray with the sonication might be able to remove these micro. Wonderful point, Matthew. Thank you. Yeah.
Oh, I think someone has added to it. Environmental factors.
Yeah, environmental factors. Yeah, they will do affect. Yeah, environmental factors will affect the attachment and detachment of microorganisms. So we probably will have to come up with an, optimal type of a processing. Yeah, definitely. Environmental factors is a point to consider. Yeah, thank you for bringing that up. Yeah, anything else? Yeah, nice discussion. Thank you. Yeah, thank you, everyone. So, and thank you, Kirat. Great job.
Hi, everyone. Hi again. I just presented and now I'll be demonstrating a really fun activity. We thought it would be really fun for people who are joining us to see if washing of melons with water is effective or not. And also if we use some antimicrobial solution to do the same is effective or not. So what we will be doing today is we have 3 melons in here. And we will be using swab method to do this. So what we will be doing is we will have 1 melon which will not be washed. The second melon will be dipped in an antimicrobial solution that I have here. So as our lab extensively works with antimicrobial and basically plant-based antimicrobials, so we have one of those antimicrobials here prepared in a bowl, and we would be practicing the immersion method of treatment with the melons today. The third melon here will be hand washed with hot water just to see if there is a difference in the microbial population between these 3 treatments. Like one will be unwashed, one will be treated with the antimicrobial, and the third will be just washed with hot water. So let me demonstrate you how I'm planning to do it. So I'll first swab the unwashed one and you can just move this bowl, and if it's visible, I have a swab here and I'll make sure that I swab all the areas of the melons. So I'm making sure that I cover most of the parts of the melons just to see if there is some growth or not. And I'll make sure that I swab the base, which is more susceptible to be in the ground. So I can dip this swab back into my test tube and I'll wait for around 10 minutes to plate this. So this was my unwashed melon, and the second one will be washing with hand. with hot water. So here it is. And we have hot water here. So we'll make sure that we, oops, sorry for that. We'll make sure that we wash it thoroughly by using extensive mechanical washing as shown to us by Dr. R in the previous demo. So I'll make sure that I get rid of all the loosely attached particles to it so that it do not contribute to the that we will see. Yep. So I think it's almost done. Here we have it, and we can swab the surface of this washed. Okay, I've made sure that the surface down because my unwashed melon has also touched the surface. So I'll be swabbing just the upper part of it, which has been washed thoroughly by me, and I will not rotate it, just to make sure that there's no contamination or cross-contamination here. So I have my swab. Yep, I think it's enough. So now we have our third melon. I'll just like dry my hands. And here is an antimicrobial solution that we generally use for all our experiments. This is a plant-based antimicrobial, and we have actually seen a very good result from these in case of leafy greens. And I personally work with food contact surfaces, so I've seen really good results with this antimicrobial solution. So today, what we would be doing is we will be immersing this melon into this antimicrobial solution for some time. I'll make sure that one side is thoroughly dipped, and we'll wait for some time. We generally wait for around 2 minutes in case of food contact surfaces, but here I think we can wait for around 1 minute just so that I can show you how I did. I did have some plates to show you, but yes, this is how I did it. So I dipped my melons into an antimicrobial solution for around 2 minutes like this. And then just making sure that nothing gets touched to the surface. And I'll swab the surface that is in contact with my antimicrobial solution. There we go. So we'll let these sit for around 10 minutes and then plate them just to see what are our results. So I do have some plates that I plated yesterday. So this is my plate from the unwashed one. I just have some condensation on the plate. And you guys can see it. So this was my plate. Not right. If it's visible, so this is from my unwashed melon that I did take swabs from my unwashed melon yesterday. And this plate is from a water washed one. So to our surprise, we did see a lot of colonies. They should not be foodborne pathogens, and they may not be foodborne pathogens. But this might be the reason behind melon spoiling really early, because we know it's a very perishable commodity. So probably these might be the spoilage microorganism which result in that. So, but if we see thoroughly, we do see that the larger colonies in the unwashed one are not visible here. So probably washing did help us get rid of some types of those microbes. And then at the end, we have the one that has been treated with an antimicrobial. So this is how the antimicrobial acts. So does it look good? Okay. Yeah. So if I see the water washed one, here is the difference. So that is how we know that our antimicrobials are really effective. And yeah. So thank you so much for patiently listening to me. And I know it's already 4. So yeah, thank you.
I just want to echo the results looks very good from the antimicrobial. I just was curious, what is the percentage of success? Is it like, I see some spots on the antimicrobial, like maybe 1 or 2.
Mm-hmm.
So what are your kind of hypothesis on that?
Are you asking how much reduction, Dr. Patel?
Yeah, because I see that it's a good reduction, but I see only 1 or 2 spots.
Okay, yes.
So
because I did not do any dilutions for it, so definitely we do see good log reductions here. But for that, I need to be sure about the number of log reductions here. I really need to calculate and have a concrete data.
Yeah, and Lynn is going to talk about it. In the next presentation, Dr. Patel will tell you what reductions, and it's one of the same antimicrobials.
Okay.
So you will see the reductions from his tables. I think the reductions are like anywhere from 3 to 5 logs. So you'll see the data in the next presentation.
okay, thank you.
And now that we just saw the demo from Kirat as to how to wash the melons with antimicrobials, and the results of antimicrobials are good. The next presentation is going to be a follow-up on that with some data. And we are going to be looking at the effectiveness of natural plant-based antimicrobials against the 2 major foodborne pathogens of concern in melons on different varieties of melons. So this presentation will be given by Libin Zhu, and Libin is the lab manager in my lab, and he's been working with plant-based antimicrobials for a long period of time. He has done several food commodities, and so he will tell us what he did and what kind of data he saw, and the answer to your question will be in this data, Dr. Patel. Yeah. Thank you. So Libin, it's all yours.
Hi everyone. So today my topic is the efficacy of plant-based antimicrobial against Salmonella, Newport, and Listeria monocytogenes on melon rind. So previous talk, my colleagues show you that there's a lot of chance when melons grow in the field, they could be get contaminated from the soil or irrigation water or wild animals. So there's a way to control to reduce the contamination is to do the post-harvest wash. So currently in the produce industry, a lot of chemical sanitizer are being used, such as chlorine, hydrogen peroxide, and PPA. But for those chemical sanitizers, because they cannot use very high concentration, so the reduction for the pathogens usually is very low, around 1 or 2 logs. So there's a need to investigate to find more options for the sanitizer. So previously in our lab, we tested some natural antimicrobial, such as essential oils and plant extracts, because they are generally recognized as safe. So when we tested these sanitizers, we found on spinach and lettuce, there's about 5 log reductions. So for this melon project, we tested a different variety of the melons, which was harvested from different states. after we receive a sample, we cut the melon rinds and then inoculate with Salmonella Newport or Listeria culture, overnight culture. The concentration is around 10 to 6 log per gram. So after the inoculation, we put these samples in the antimicrobial solution for 2 minutes wash. After the washing, we put the samples in the fridge, and then we do the enumeration for the survivals on the day 0 and day 3. We have a lot of data, but we don't have enough time to show it today. So I just pick some samples to show you. So these samples are from North Carolina. These are different varieties. So we use the PBS as a control. For the plant-based antimicrobial, we use the 5% olive oil extract and 0.5% oregano oil solution. From this table, you can see on the day 3, all the Salmonella Newport, they are under the detection limit. So it's around 3 or 4 log reduction on Salmonella. Also, we can see that even on the day 0, on some varieties, the Salmonella was also below the detection limit. Also, here's a table for the Listeria reduction, survival of Listeria on the melon rinds after the treatment. Similar to the Salmonella reduction, we can see at day 3. For many of the varieties, we couldn't find any survival Listeria. So in summary, compared to the control, the plant-based antimicrobials caused around up to 4 log reductions in the population of Salmonella and Listeria. In general, the oregano oil had better activity than the olive oil extract. Also, from the 2 tables, we can see the antimicrobials were more effective on Salmonella than on Listeria. Also, we saw the antimicrobial had better reduction on honeydews than on cantaloupes. Also, we saw the antimicrobial were equally effective on different variety and hybrids. So from this project, we can see the plant-based antimicrobials have the potential to be used as a sanitizer for the decontaminating melons. Also, these measures will help improve the safety and the shelf life for the melon. Okay, I would like to thank all the collaborators, also USDA fundings for this research. So any questions?
Thank you, Libin. Any questions for Libin?
Yeah, while we're at it, when you were showing the charts, there was a Da Vinci further back. No, that's it. No, I think it's a previous one. Yeah. So here, it's starting off with a control of 4.72. There's a little higher control there, but it only goes down to 2.85 or 2.9. And I guess the question is, why does it stay that high? Are you finding anything?
Yeah, the question is for Da Vinci variety. Why the Listeria population are very high even after the treatment? Yeah. So the reason is because for the Da Vinci, this variety, the night they have the night density are very high. Also, the deeper because yeah, the mainly because of the night structure. Yeah, it's more dense than other varieties. Yeah.
So it's the density of the net structure.
Yeah.
Now, and then you, of course, they do scanning electron microscopy, so you see what it looks like.
Yes, I'm not sure if we have that picture here. Yeah, some collaborator in Texas, they have the pictures of that night. Yeah.
Thank you.
Okay, thank you.
Any other questions for Libin? That's a very good question, Matt.
Thank you.
Yeah, if you see Da Vinci, it's like really naked. Yeah.
Well, you look at any Da Vinci.
So any questions from the online?
I just have a small question, Dr. Sadhana. It's a great work. I was just curious to know the detection limit was 1 log CFU per gram, right? So in order to answer Dr. Patil's question, we can definitely say in a plate, how many colonies did you observe in an undiluted plate in order to tell it's 1 log? That will answer the question, I guess.
Sorry, can you repeat? What's your question?
So for the detection limit as a 1 log CFU per gram, how many colonies did you see on the plate?
1 colony.
That's it. Yeah. That's what I just want to emphasize over Dr. Patil's question. So.
All right.
Okay.
Yeah, thank you, Libin. Any other question from the audience here? Yeah. Oh, somebody has a question.
For like a comment, it's related with the Sakara variety. So the seed plant cannot reach the surface and kill the bacteria because of the surface. So if we should expect more outbreaks on this kind of variety or no? Not necessarily.
You mean the Da Vinci Sakata, more outbreaks on the particular variety? Yeah.
Is it hard to disinfect because of the net is different and probably the structure to protect the bacteria to the resistant region? So I don't know if it could be more outbreaks of these pathogens and in this Da Vinci Sakata variety or not?
So the question is, have you seen any information on the outbreak is Da Vinci Sakata involved in outbreaks is what it's asking.
No, because usually when they report the outbreak, they don't mention what's the variety.
They usually don't mention what variety it is.
Yeah.
But I suspect for the way that is netted, it is always possible. Yeah. We probably have to do more research into it, but we only find out what the organism was who was affected, but they never mention the name of the variety. Yeah, it's a difficult one to say whether there was an outbreak involved in that or not. Yeah, absolutely. Yeah.
Okay.
Anything else?
I have a question, Dr. Ju. That's a good presentation. So what do you think? I think there is a question also from the audience. There was a very good question about why Da Vinci is different than rest of them. So I think that will probably trigger us a lot of future opportunities for research. So I was just thinking like maybe Da Vinci, which is really a very well-known variety from the company, that's why they're making a lot of money. And it's also very sweet melon. It's really marketed very well compared to other varieties. So I was just curious from this research, do you have any specific hypothesis on some of these antimicrobials, how they act on skin? That's how it is that the log reduction is going down. Then we can start thinking about some of the characteristics of the particular compounds in melons. And I'm suspecting the antimicrobial compounds we measured on the Da Vinci could be lower than some of these hybrids. I had to go back and check again. I think Dr. Singh is not there on the Zoom now, but I think Da Vinci has a little bit lower antimicrobial compounds, but more sugar. That's why a lot of consumers like that melon. I was just curious about the hypothesis or the mechanism of action of some of these antimicrobial compounds.
Yeah, the mechanism is because those natural antimicrobials, they are polyphenols. Usually they can disrupt the cell walls.
Okay.
But for the Da Vinci, because if the bacteria attach it and internalize too deep inside the rind, then the antimicrobial solution is very hard to reach the cells.
Okay.
So,
yeah, that sounds really good answer. So, but I was just curious also, is there anything related to higher sugars? And like a lot of this particular Da Vinci is very sweet and people like it, but in the same variety, we didn't find a lot of antimicrobial property compounds in the skin, like when we did the volatiles. But again, I had to recheck with my postdoc the real values. I don't remember exactly offhand.
okay, thank you.
Mm-hmm. Yeah, and I think maybe Dr. Ravishankar and all will discuss about that. That's an interesting observation you're making. I think it's a good point the audience raised that triggers us some new thoughts about that.
Yeah.
Thanks. I think it's Matt, right?
Yeah.
Is it Matt is the one who asked the question? Thanks, Matt, for that.
Yeah, absolutely. We can definitely look at that, Dr. Patel. We probably should research this Da Vinci saccata a little bit more. Because it is hard to inactivate the bacteria on the surface, and again doing some of the correlation work will tell us what type of volatiles are in there and are they playing a role in this. We don't know any of that yet. But as Libin mentioned, what we are working with is some of the polyphenolic compounds and some of the antimicrobials. It is very well known that they interact with the bacterial membrane. So that much we definitely know. Yeah. But what is exactly happening, at the surface netting level is something, beyond the scope of this work. We haven't done that yet. Yeah. So there is one more question here. How was the attachment strength on Da Vinci? We have to go back and check. Yeah, that's something that we will have to look at it. Yeah, I can't remember, off the top of my head. Yeah, but definitely, we'll definitely take a look at that attachment strength on Da Vinci and try to correlate that along with everything else involved in the project and efficacy of antimicrobial. So a lot of the correlation work definitely needs to be done. So yeah, we'll look at that too, Nitin. Thank you. Any other question? Yeah, very nice discussion. Thank you, everyone. So thank you, Libin. We'll I know in the interest of time, we'll keep moving. Yeah, great job. So so far we were looking at the food safety aspects of melons, and we saw a nice story all the way from the field to post harvest what is happening. Now we are going to switch gears and look at shelf life of melons. So the next couple of presentations are going to be looking at the background microbiota as well as microbiome. Is what we are going to move on to. So the next presentation is on some of the work that we did looking at the initial background microbiota. We began looking at the shelf life, but the results were very inconclusive. So we moved into looking at what is the initial background microbiota and how can we predict the shelf life based on that. And the presenter is Elizabeth Sargent. Elizabeth is a graduate student in my lab from the Applied Biosciences Graduate Interdisciplinary Program, and she's been in our lab for more than a semester now. And she's been looking at a lot of the background microbiota work on different food commodities, including melons as well as leafy greens. And now she's going to tell us about what they did with regard to background microbiota and give you a little bit of an example of the results that we have. I mean, it's a lot of data, but just to save time, she's going to show us just representative data probably from 1 year. So it's all yours, Elizabeth. Thank you.
Hi, everyone. Thank you, Dr. R, for the introduction. As Dr. R said, I'm a graduate student in her lab, and I'll be discussing the comparison of initial background microbiota on the surface of melons grown in different regions in the United States. So to begin, I'll do a brief introduction on our ideas behind this like mini sub-project of the overall melon project. I'll talk about the objective we had with the project, go over the methods, then a brief data and results for one of the years, and summary and conclusions, as well as general acknowledgements of our collaborators. So to begin, melons, as we know, are perishable and known to spoil. And we know that many physical, chemical, and microbial aspects can contribute to the shelf life and the spoilage of melons over time. So to develop ways of extending shelf life and potentially preserving the quality of the melons, we evaluated the initial background microbiota that survive and even grow on the surface of the melons prior to them spoiling. So the objective was to grow and count and then compare the initial background microbiota load on the melons that were grown in a variety of different regions in the United States. And we also hypothesized that the melons that had low initial background microbiota may have a higher shelf life in comparison to the melons with high initial microbiota load, which would have a lower shelf life. So the methods. So to begin, we took rinds from 2 of the melons of each variety. Then we took the rind, measured out 10-gram squares of it, and that was all sterilized using our aseptic technique. Then we placed the melons into a 90 milliliters of buffered peptone water and then stomached for 2 minutes. And then once we had that solution, we took those the microbes that were then in the solution, extracted them and made serial dilutions, and then we diluted those samples and then spread them onto TSA plates, which were then incubated at 37 degrees Celsius. And after 1 to 2 days, they were then counted, and that's how we gathered our data. So table 1 is comparing the initial background microbiota load between the selected varieties grown in different regions. So here, Arizona actually had the lowest initial background of 5.52 log CFU per mil, whereas North Carolina had the highest log CFU per mil at 7.57. So then to the far right column is our averages of the different varieties in log CFU per mil. So you can see that the highest variety TH5 had the highest log CFU per mil of 7.61. Then variety TH6 had the second highest, moving to TH13, which was the third highest at 6.84. And then our lowest log CFU per mil number was 5.52 for the variety TH16. So as you can see here, this is the same data set, just displayed in a graph form. And it's also showing the full range of the log CFU per mil microbiota data results that we got. So you can see that Arizona and California had the largest range here. However, North Carolina had the highest initial microbiota background. So in summary and conclusion, to determine the initial background microbiota of melons allows us to understand the microbial quality and what may affect the shelf life of the melons. The environment where the melon has been grown could significantly affect the initial microbiota background. Melons grown in environments of high heat, low humidity had a lower initial background microbiota, which may have a longer shelf life. And melons grown in environments of low heat, high humidity seem to have a high initial background microbiota load, which may contribute to a shorter shelf life. So this may influence future research and understanding the microbiota load and of a melon and shelf life needs to be evaluated further in the future. So these are some references of the papers that we use to help support our data and our findings, as well as the acknowledgement. I'd like to thank the Yuma Center of Excellence of Desert Agriculture, Texas A&M AgriLife Research, the USDA National Center of Excellence, Vegetable and Fruit Improvement Center, Foods for Health, and the Animal and Comparative Biomedical Sciences, as well as Dr. Ravishankar and Libin for giving me this opportunity, and the rest of the volunteers and contributors to these projects. So thank you. I'll be taking questions.
I think there may be a
Okay.
question on the chat.
What background microbiota did you look at? So the question was what background microbiota we looked at, and then she further asked if it was total coliforms. So we actually played it on TSA, which is tryptic soy agar, which basically gives the nutrients to all bacteria. So we were just looking at total bacteria on the melon, on the background of the melon. No, we did not. Oh, HPC. Yes, yes, we did. Yes, sir.
It's all microorganisms.
Yes, all microorganisms. Yes. Thank you.
So this is again Bimu Patil from Texas A&M. It's an interesting study. I like that the outcomes. When I say I like it, it's an interesting outcome which we may not like it. The reason is because we are finding this TH6 is highest load compared to the ones which we didn't like. Some of the hybrids, like 16 is lowest load. So that means the higher the load microbes, that means the storage life is not good, right?
Yes.
So that means TH6 will not have good shelf life.
Can you say that one more time? I'm sorry.
The TH6, which you have showed that it has higher microbial load, that means it may not have good shelf life then, right?
Yes.
Okay. So that's an interesting outcome, I think, which we need to address, and I don't know how do we address it, because we are considering TH6 is the best variety so far. Again, we didn't do specific study on TH6 yet, on the post-harvest shelf life. We need to check on that.
Yes. One thing we didn't consider in this part of the sub-study was the physical or the chemical aspects of the melon. So that could also contribute to the shelf life. So that wouldn't need further evaluation. We just evaluated the initial background microbiota, which may contribute to a lower shelf life if it is higher. But we still need to gather further data on that.
Okay. It's only one of the factors.
Yeah, it's only one of the factors.
And it's just a prediction, and we probably need to do further studies on this, Dr. Patil, because for all we may know, this is like the total microorganism count on there. So what if it is a lot of it is probiotic? We don't know that. I mean, we have to do a lot more further study. So it may be beneficial bacteria, it may be spoilage bacteria, we don't know, but it's just a prediction. And it's one of the factors. So it's a very small part of the very big picture. And we just, we just wanted to see how the initial, microbiota load, what the initial microbiota load is on, different varieties. So I think further study will need to be done on TH6 to see, what they are, and maybe, some identifications probably need to be done. Yeah.
Okay. Yeah. Thank you.
So now we are going to look at microbiome studies. The speaker is Madison Goforth from Dr. Kerry Cooper's lab. Madison was one of the very, very first volunteers that worked in the Melons project starting 2018 when we began the Melons project, actually. She helped out in our lab and later on moved to Dr. Kerry Cooper's lab, and she has been looking at the melon microbiome. So she is going to tell us about what kind of diversity, microbial diversity she has been seeing in the different varieties of melons that have been grown in different regions. So Madison, it's all yours. Thank you.
Hello, everyone. Welcome back. So yeah, like Dr. R was mentioning, I've been working on this melon microbiome for quite a bit, and I'm now coming towards the end of the analysis in terms of generating alpha and beta diversity figures. So a little bit about the introduction on this melon microbiome that I've been trying to look at. Little is known about the microbiome of melons. There has only been one published paper out there looking at cantaloupes, only one variety in Mexico, and as well as the microbiome on the hands of the workers. So that is pretty interesting data. We didn't actually look at the workers' hand microbiota. However, being able to look and explore multiple varieties of melons as well as regions will help explore maybe more of a standard set. But there is good understanding to this microbiome that could help with combating modern bacterial issues with melons, such as pathogenic bacteria and how antagonistic or protagonistic bacteria can play a role in such foodborne pathogens. And in later terms can help improve food safety concerns, develop roles for antimicrobial resistant bacteria, and look at how gene transfer and modification of certain bacteria could play a role in future growth of melons. So a little bit of the methods that we were able to do from being able to collect melons from Dr. Ravishankar's lab, we swabbed a 5 by 5 centimeter area with a microbial enzyme solution that allowed for the DNA not to be sheared. And so what we ended up happening over the past 4 years, we collected around 610 samples from 7 locations, 6 states such as California, Arizona, North Carolina, Georgia, Indiana, and Texas. But then in the next step, we DNA extracted and amplified the V3, V4 region for 515, 926 primers. Lastly, we sequenced with Illumina MiSeq and then analyzed using Chime2 Linux. So a little bit of difference between alpha and beta diversity, just some different languages that we use. For alpha diversity, we're comparing melon to melon grown within the same field. So if Arizona has Da Vinci as well as F39, we're going to be comparing those two. Such as versus beta diversity is looking at melon to melon in different fields, so that F39 compared from California to Arizona. So first up, we actually had 3 different types of melon microbiotas to look at. We did a harvest melons, a specialty melons, and a commercial melons. Harvest melons, which was where we as a lab were able to go directly to the field, agricultural field, and collect different samples. These are the same samples that we did use for the prevalence, except for we collected separately. So for the harvest melons, looking at alpha diversity, I just decided to give you guys a bit of a difference between sample types of melon versus soil microbiome. As you can see for Shannon diversity, what that is indicating is the evenness or phylogenetic tree branching off of the bacterial communities within melon and soil samples. And for the p-value, as you can see, it's pretty low. So meaning there's a statistical significance between the evenness of bacterial communities. Next is biodiversity. So again, this is looking at sample types within the different sites. So there's a lot of clumping of different sites. What's really interesting with the melon sample types is all from all 4 locations: Arizona, Huron, Imperial, and Yuma. So Arizona and Yuma are like pretty much grouped, while Huron and Imperial are a little further. They're all pretty much grouped together for melon sample types, meaning bacterial communities are very similar to one another. However, with soil types, what's really interesting is Yuma compared to some of our other Arizona samples are pretty further dispersed. We did take them slightly at different points of the same year though. So for our harvest melons, we collected from May of 2019 to August of 2019. Next, I just want to point out this heat map of about 20 actually bacterial families between melon and soil. So at abundance, when it's a lot more redder or darker in color, meaning that there is more of abundance of that certain bacterial family. What's interesting here is the 2 species at the top for melon are more prevalent within that subset of samples than it is in soil, as well as just if you go further down, there's like white spaces, meaning that there's not really much of a presence of those communities. Our next category is specialty melons. So again, Shannon diversity is also looking at melon type. What was interesting is that for Shannon diversity, there's a more statistical significance for melon type varieties than there were for the regions that these melons were grown in, which makes sense a little bit. So what we have from left to right are the varieties, and the dots are just representing outliers, but there is a little bit more of a diverse branching in certain melon types between like the SSAs all the way to STAs, with SSU being a little bit higher up within the branching of their bacterial communities. Next, for their Bray-Curtis plot, as you can see off to the right, there are groupings by the regions that they're grown in. So obviously Texas Uvalde and Texas West Silico are from the same state. However, they're different fields that samples were collected in. So there's a little bit of some grouping and varying in bacterial communities. However, what's really interesting to mostly pinpoint is that Arizona and California is a very wide, very broad distinction between the rest of the groups. They're a lot more jam-packed. Next is commercial melons. So these melons, oh, I forgot a little bit. So harvest melons we collected straight from the field. Specialty melons we got from Dr. Ravishankar. And then lastly, our commercial melons were taken straight from stores around the Tucson, Arizona region, from Bash's, Safeway, Natural Foods, or Whole Foods, Natural Grocers, all those other little niche supermarkets. And one of the things that we've looked at were taking organic versus conventional grown melons. Down at the bottom is the melon type, and within the parentheses are the number of melons that were actually sampled. And again, for Shannon diversity, there's no difference between bacterial communities or evenness or branching, sorry, of those bacterial communities between organic and conventionally grown. However, just remember that these are after going through a certain food systems process. And then lastly, I also wanted to pinpoint also how it was different between melon types. So even though they did conventional and organic, melon types such as cantaloupe, honeydew, miscellaneous, and watermelons did show actually statistical significance between that branching of bacterial communities. So onto the Bray-Curtis plot, or the beta diversity. For just the conventional, high in brown and organic in purple, there doesn't seem to be much grouping necessarily, and it seems that there's no real difference with bacterial communities. So a good summary For harvest melons, they showed high branching of bacterial communities, as we had seen between melon and soil types. Transference of soil microbes onto melons seems to be seen at a lower abundance. So as I was showing you with that heat map, there seems to be some less bacterial communities in that soil versus on the melon and vice versa. Specialty melons also differed in bacterial communities based on type of melon for alpha diversity. However, for beta diversity, it depended on regionality. And lastly, commercial melons did not differ in evenness of communities, nor did they show bacterial community similarities for conventional or organic. And diversity seems to decrease through the food handling process. I haven't shown you guys the taxa map for harvest, commercial, or specialty, but for reference, I was able to see observed species between 300 to 400 species for melon and soil types in the harvest section, while for the specialty melons, it was a log reduction. And then for commercial melons, it seemed to be about 2 to 3 log reduction, only about like 2 to 5 species at max for the taxa plots. But then for some future directions, I'd like to see is studying protoxic and antitoxic bacteria microbes in relation to foodborne pathogens, like I was saying in the introduction, to better develop HACCP protocols if it needs to be based on the melon type or regionality, and prevent future outbreaks from analysis of previous years would be helpful. Lastly, understanding components of the microbial community that could grow a better melon and increase crop yield based off of soil microbiome or the melon. And these are some other points in just discovering potential genetic modification of microbes based on plant growth or interference that could possibly show a relevant point of view for future growing seasons, and then decreasing potential antibiotic resistant microbes is always best. Lastly, I just want to acknowledge the ACBS Center as well as the Yuma Center of Excellence for Desert Agriculture and all the grants that were able to fund this project. Thank you.
So I have one question.
Yeah.
Madison. You mentioned the commercial melons, right?
Mm-hmm.
So what was the sample size there for the organic and conventional ones? That's very
Yeah.
interesting to me.
So what was interesting for the conventional, we sampled around 149 melons. And then for organic, we sampled only 88. But what was interesting from our sampling methods compared to harvest and specialty, we actually washed the entire melon to get that microbiome off of it instead of just the 5x5 region.
Okay, so the entire melon was sampled.
Yeah.
And was it all cantaloupes, or did you include different varieties?
Yeah. So we did cantaloupe, honeydew, and watermelons. There was also miscellaneous with 45 sample types, but everything else was pretty dispersed, with cantaloupes being the highest at 95 samples.
Oh, okay.
Yeah.
Thank you.
Cool. Yeah. Thank you.
Now, we are going to be moving on so the research part is done now. So thank you. And we are going to be moving on to talking about some outreach activities. So over the years, we have conducted a number of different outreach activities at the University of Arizona here. And you did hear about one of the major outreach activities that were done in Yuma. Martin did mention about the field day. But we have been working with school students, a number of other presentations we have been doing. So we wanted to just highlight some examples of these outreach activities. And since Richard has been has played a major role in all of these outreach activities, he is going to be presenting to you as to what we have done. Just that's why it says selected, because we have a lot. Just in the interest of time, he's going to tell you what we did with the school students as well as with regard to presentations and conferences. So it's all yours.
Thank you, Dr. Ravishankar. So again, like she said, we're only going to cover a small select snippet of all the outreach activities that we have performed over these 4 years. So one of the first ones I want to talk about is that we participated in the Southern Arizona Research, Science, and Engineering Foundation, also known as SARSEF here. And we specifically, for this instance, participated in Future Innovators Night in 2019. So SARFSEF usually holds its conference and science fair at the Tucson Convention Center. And their science fair, they do every year from pre-K students upwards to 12th graders, so seniors in high schools, from students all across Southern Arizona. And in this science fair, they have over 2,000 projects compete for the Regional Science and Engineering Fair. And what Future Innovators Night is usually after the science fair, where kids, even adults, parents, high school students, college students can get hands-on science activities, mingle with professional scientists and engineers, and celebrate their research conducted by thousands of Arizona students each year as well, looking at their posters and going to the different booths of the different businesses throughout Southern Arizona. And so what we did was we showcased the importance of the food safety and microbiology work, also the prevention of cross-contamination and use of natural sanitizer for washing produce, and ongoing food safety melon research work done in the lab. So the attendees are around thousands of children and their parents visiting all the science fair posters and exhibits. Another one that we participate in is the USDA Ag Discovery Camp, where students visit from all across the country, including its territories. So we participated in this event since 2018, including this year in June. And this is a unique opportunity for students to explore agricultural sciences, where they can gain knowledge on these careers, and whether it be animal, plant discipline, wildlife management, veterinary medicine, biotech, entomology, food safety, food production, and so forth. And here students get to experience hands-on learning workshops here at the U of A, behind the sciences tours, laboratory tours, even field exercises across Arizona, and other activities. And here we demonstrated the ongoing food safety work, specifically the Melons Research Project, and other research projects performed in Dr. Ravishankar's lab. Over the course of the 4 years, we had probably around over 60 high school students. Again, this is not just in Arizona, but across the US and its territories. And also the mentors that come also get to participate in these activities. In terms of a U of A project is the Safer Food Cats High School Summer Program. And we participated in 2019, 2021, and 2022. I believe 2020, it was not performed due to COVID. And here, these students high school students under 18 get to experience a week-long tour here at the University of Arizona, where students get to introduce to the science that keeps food safe. Specifically, this is a food safety program where students get to learn, even practice techniques used in real-world food safety laboratories at the U of A. Here, what they get to do is they're introduced to the campus, wildlife heritage, they get to engage on hands-on STEM activities, tour food safety research labs all across campus, participate in field trips, and listen to food safety researchers and professionals. In this program, we probably interacted with over 20 high school students over the course of the three times that we did this. And moving on is the Southwest Ag Summit in Yuma, Arizona. We were fortunate enough to be able to present posters in 2020 and 2022. And what the Southwest Ag Summit is the premier agricultural show in the Southwest. Apart from continuing education opportunities, what the Southwest Ag Summit provides is opportunities for people to learn about what others do in the industry and ways that people can work together. The target audience for this one is usually agricultural professionals from Arizona, New Mexico, Southern California, and Northern Mexico. And over the 2 times that we've done this, we've presented 5 poster presentations. We also are going to participate in the International Association for Fruit Protection annual meeting. So we did it in 2019, 2020, 2021, we didn't participate so much due to COVID concerns. However, this upcoming one in 2022 and next week in July 31st to August 3rd, we will be heading to Pittsburgh to present some of the research we did in the Melons project. Here at the IAFP annual meeting, attendees get to get information on current and emerging food safety issues, the latest science, there's many talks, innovative solutions to new and reoccurring problems, there's breakout sessions and rooms, and opportunities to network with thousands of food safety professionals from all across the world. Here we have presented 6 poster presentations in the 2 years. However, this upcoming one, we are presenting 2 posters on the melons research we have done. We also have participated in the annual Food Safety Conference. This is hosted by the University Food Safety Consortium, which Dr. Ravishankar is the chair of. And we participated since its revival of the annual, both the Food Safety Consortium and the annual Food Safety Conference in 2019 upwards to 2021. We will participate in the 2022 one, which happens sometime around October. Basically, what the University of Arizona Food Safety Consortium is, consists of members from across the University of Arizona, as well as members from the industry, the state of Arizona Department of Agriculture, and state health agencies. And here, the main focus of the Food Safety Consortium and the conference is to promote networking and provide educational opportunities to its members and others. And also, we host the annual Food Safety Conference, and there we can reach a wider variety of speakers. So far, the first year we've done it was in person, but then again, due to COVID, it was online. Hopefully this upcoming one will either be hybrid or online and in person. And so far we presented 6 poster presentations. We've also done 2 oral presentations at the meeting. So apart from that, those are only, again, the small snippets of outreach activities that we have done. There's a lot more, a very long list. And here I'd like to thank everyone at the University of Arizona and Dr. Sadhana Ravishankar for giving me the opportunity to work on this project for the past 4 years. And Libin Zhu, the lab manager, who has provided me great insight on how to go through with this project, along with the former members of the lab and current members, and also the Yuma Center of Excellence for Desert Agriculture, like Martin, who helped bring the melons all the way from Tucson here, including those from Texas A&M, such as Dr. Patil, for letting us be involved in this project. And of course, the funding from the USDA.
So thanks, Richard. And I know we are running out of time, so I'm going to be really quick. So mine is the last presentation. And I just, before I went on to, closing remarks, I just wanted to talk to you about some of the testing services we can do at the University of Arizona. And so it's going to be a real quick talk. The content of my talk is going to be, I'll just give the list of what kind of testing analysis we can provide to the food industry. And I'll give a very brief description. And even though I say, discuss what HACCP is, I'm sure, everybody knows about HACCP. So I'm not going to be spending a lot of time in there, and, why HACCP is important, and, what we can do to help you with regard to developing HACCP plans. So this list given in here can tell you what type of, testing we have been doing. So we can do shelf life testing. We can test for many different types of microorganisms, indicator bacteria, E. coli, coliforms. A lot of these came up today. We can also test for foodborne pathogenic bacteria, including the major ones, E. coli 015787, Salmonella, Listeria, Campylobacter, Bacillus, Conobacter, you name it, any of these. We can test from food products as well as environmental samples. We can also do food quality testing, like texture, color, viscosity. We have equipment to do all that kind of testing. And another one is we have an aflatoxin analyzer. We got it out of a grant when working with a nonprofit organization. And aflatoxin is a carcinogenic compound that can be present in many different types of food products. And we have in Arizona specifically mesquite. So mesquite flower is used by many of the small growers in here. And because of high humidity conditions, sometime in the summer, like, we have monsoon now in July, there is a concern about mold growth and aflatoxin. So we do a number of mesquite flower samples in our lab with small growers. And we also have some general public coming and testing things with us, like acorn flower. There was a gentleman who was using acorn flower in his breakfast. cereal, and he had a concern about aflatoxin. We have tested coffee, many different types of food products. We can also develop HACCP plans for the food industry. Anyone that is, a new startup wants to develop a HACCP plan, we can do that. And some people even, have a developed HACCP plan. They bring it to us and we review it for them. So we can do that as well. We can look at the efficacy of sanitizer, and we work with a lot of sanitizer companies to validate their product in food type of applications. So we can do that. And then finally, we can also do some sensory evaluation in collaboration with the nutritional sciences, and in their labs, there are sensory booths. So we could do that type of testing as well. So now I'll go a little bit more detail. So for shelf life testing, we have done all different types of food products. In the past several years, we've been doing this. Libin is the one who does, all this work. So you can see the list of different types of food products we have done the shelf life testing on. And then microbiological testing, you can see the list again: indicator bacteria, aerobic plate count, coliforms, generic E. coli. We can also do lactic acid bacteria, because oftentimes people want to know what type of probiotics may be in their food products. So we have done that. Yeast, molds, and the list can continue. And foodborne pathogens, the big list is already there, and I already told you about that. So you can look at that list as well. Efficacy, again, we can do before and after. So we can do a control and then treat them with the sanitizers, and look at the specific microorganisms to check what kind of effectiveness these sanitizers have. And antimicrobial effectiveness testing, definitely, we can look at either the inherent antimicrobial properties, or we can do like an inoculated type of study where we would be able to add different concentrations of the antimicrobial and see if there is a concentration dependent or time dependent type of an activity. And for food quality, pH, water activity, we have a moisture analyzer. We have chromometer to look at the color, texture, viscosity, and so on. Aflatoxin testing, you can see the different types of products we have tested, aflatoxin on mesquite flour, corn flour, spices, cereal products, and so on. And then we can, do HACCP plans and sensory evaluation. So HACCP plan, in the interest of time, I'm not going to go too much into detail. It's a prevention based program that the food industry has been using to minimize, food safety type of hazards, both physical, chemical, and biological, all different types of hazards, and produce a safe food. And HACCP originally was developed by NASA for one of their programs, and then it was slowly adapted into the food industry several years later. And HACCP applies to both domestic as well as imported type of food products. And by using HACCP, the food industry has been, clearly able to show that there is a reduction, at least 30% reduction in foodborne illnesses due to meat and poultry type of food products. And that's where HACCP was mandated initially. And on to your left, what you're seeing is a little HACCP flowchart developed for a minimally processed vegetable products such as baby cut carrots. And HACCP works by, looking at what are the critical control points, establishing critical limits, and finding ways to monitor, coming up with corrective action, and verifying everything, and record keeping, and so on and so forth. So there are seven different principles of hazard of HACCP. One can conduct a hazard analysis, figure out what are the critical control points where you cannot deviate too much, and then establish critical limits for these critical control points which you cannot go beyond. And then you can monitor, establish corrective action, verify whether everything is functioning properly, and keep a good record keeping. So that's what HACCP is about. So we can develop HACCP plan, and this is something we always use in our reports to the food industry. Just a disclaimer: we try to do everything very, very carefully, but we like to keep this. And this is our website. And we recently launched a new website just for our food testing, which is foodtesting.arizona.edu. It looks something like this. It was designed by Elizabeth Sargent. Thanks to you, Elizabeth. She did a great job in designing this website from all the way from scratch. And we just launched it. So this is our contact if you want to do any type of food testing. And we also have an email, which we just got for food testing, acbs.foodtesting / foodtesting@arizona.edu. So, if anybody is interested, please check it out. So that's on the food testing I have. Does anybody have any questions?
I have a question.
Yes.
Can we extend these services beyond Arizona to like?
Oh, yeah.
Yeah.
Yeah, we have worked with the companies beyond Arizona as well. Yeah, people from the East Coast, we work with sanitizer companies on the East Coast. And I remember Libin did testing for pet food one time, and that came from the East Coast too. And we even did some, chemical testing for them. Yeah. So this is nationally, and we have done internationally too. We have worked with a company in Mexico to find application for their sanitizer in the food industry. Yeah, so we can work with anyone. Yeah, that's a great question. Thank you. The question was, can we work outside Arizona? And the answer is yes. Yeah, we can do it with any company. Yeah. Thank you so much. So now this brings us to the closing remarks for the meeting today. And before we finish, I just wanted to show the participants online that we prepared like a 2-pocket folder for everyone that is attending in person. And we included a bunch of the information about the project. So we will try, and make a PDF of all this and try to, send it to the online attendees. We have an introductory material. We did, we have a 2-pager from YCDA, giving a summary of their field growing as well as outreach activities. We have a handout, a 1-pager showing a summary of activities with regard to food safety at the University of Arizona. And then we have again 4-page activity with regard to outreach, 4-pager with the selected outreach activities, and then a list of food testing. And then we also have a trifold brochure that we created. Kirat and Elizabeth created this, showing health facts and fun facts about cantaloupes, and also different ways to use cantaloupes in recipes and so on. So we have a trifold brochure. And they also came up with a new recipe, which we tried in the lab today and gave it to all the participants. It's called the Perfect Cantaloupe Smoothie. So we have included that as well. And then we have included 5 different papers that came out of the research in the Melons project, 3 from Dr. Patil's lab and 2 from our lab. So we do have this. I just wanted to share that with you. Hopefully we can put PDFs of all these for you sometime. So they say it takes a village to raise a child. So this workshop was truly a team effort between the various institutions, and mainly, we did a lot of the effort was here, but then also we got the cooperation from Texas A&M as well as the Yuma Center of Excellence for Desert Agriculture. So, and I would like to thank everyone. First of all, I would like to thank all the presenters for taking time out of your busy schedule to come and talk to us all about that. Thank you so much to all the presenters, both from Texas as well as from Arizona, Dr. Crosby, Dr. Patil, Dr. Singh, Martin from YCDA, and all my team members for preparing all these presentations and taking the time to talk to all of us. Thank you so much. And I would definitely like to thank my team pictured in here. This was the team that put everything together here. And I would first of all like to thank Richard Park. He was the coordinator and organizer of this conference. He prepared the save the date, came up, we came up with the schedule. He helped prepare a bunch of the handouts, set up the Zoom link, and looked at all the registrants. And, drafted the email for me yesterday, and, which I was able to send to all of you. Helped us with all the technology and, doing demonstrations as well. So thank you, Richard. A big thanks to Richard for, basically, wonderfully organizing this conference. I would also like to, and Richard is pictured in here, and I would also like to thank graduate students, Elizabeth, Kirat, as well as Alhan. All three of them are in here. They did all the work in the lab, setting up, helping us set up the demos. They prepared the, some of the handouts, went to Kinko's to print those out, and they were like, okay, we can do this. We can, do some recipes. We can prepare smoothies. So a lot of the work has been going on into that too. So thank you, girls, graduate students. Thank you so much. And Libin for, cleaning up the lab and, getting it ready. I know Libin has been busy doing out of the industry work, but he's been, able to help in his capacity. So thank you, Libin, as well. And thank you to all the other researchers who have, who are pictured in here, who contributed to, all the work we presented today. A bunch of them are in here, including Madison, who presented the work from Dr. Cooper's lab here today. So thank you, all of you. Thank you. And I would like to thank Texas A&M team, Dr. Patel, Dr. Singh, for sending us all the information. I kept asking them for the information. I'll be like, I need this, I need that. So I think they kept sending all the information to us and, for presenting today. Thank you. And his, lab team is pictured here as well. And then I would like to thank the YCDA, specifically Sonat, Martin, and Paul for also discussing all these activities with us. And they also, some of their outreach funding came towards to support this particular workshop. And Sonat was, constantly involved in the preparation and the, schedule and so on. And, they sent Martin here to deliver the speech about the growing and so on. So a big thanks to them. And Martin, thank you so much for patiently bringing all those melons every time we needed to, for us to do the research. Really appreciate your help. I know you've been like so helpful. We didn't have to go and harvest these, melons ourselves in the hot fields and so on. So thank you. Thank you, Marty. And I would like to thank Rosa Bevington. She is the media person. She took a video of the entire program that happened in the morning, the in-person demonstrations and the workshop. So she traveled all the way from Yuma to do this for us, and she had to leave. So thank you, Rosa. And we'll be sending those, videos to Dr. Patel and everybody else involved. And again, thank you all the presenters. And I would like to thank everyone that helped distribute the information, all the club presidents, all the student club presidents, all the program and academic coordinators, and some of the colleagues who helped disseminate the information. And Sona did the same thing too. She was able to send it to everybody, all the growers in Yuma. Thank you for that. And thank you, Dr. Patel, for sending it to all the project collaborators and teams. And that really helped us in, getting a lot of the audience. So a big thanks for that, to help distribute the information. And last but not the least, I want to thank all the audience, all the in-person attendees for taking time out of your busy schedule to come in person, and for everyone that is present online. I know for those in the East Coast as well as in the Midwest, it's beyond 5 o'clock. So thank you for patiently listening to, all of us, in this workshop. Thank you so much. And finally, yeah, thank you, Penny. Penny is right here. Yeah, she came with Matt. Thank you, Penny, for being patiently listening to us and being quiet here. Thank you. So big thanks to USDA-NIFA Specialty Crops Research Initiative, which is the Melons project through the National Center of Excellence for Melon at the Vegetable and Fruit Improvement Center at Texas A&M. Without the support from the USDA, we would not have been able to do this project. So thank you so much. all the financial support we have here. And so this concludes the workshop today. And I know, big, great team, thank you. So there is, I think, really good comments in here. Yeah, thank you all. And I hope you all had a great experience, a great learning experience. And I think, ours is a great team, and we will continue to do, other projects as well. Hope we get a chance to collaborate in future. So thank you, everybody. So this concludes the session today. And we'll try to reach some of these materials in the handout to you with the permission, from the PI. Yeah, thank you.